Welcome back, and thank you for being with us today again. And we welcome you back and hope you were well rested. And after you got some rest, hope you managed to read up all those 3 articles and the entire handbook that we gave you yesterday, about 65, 70 pages. I'm just joking. Obviously, you didn't read it in 1 week, so obviously you wouldn't be able to do that overnight. But I hope some of you did manage to read some of the abstracts so that whatever we continue today, it's all going to be related to the same 3 articles. Nothing out of those 3 articles which be in detail. We may allude to a few examples, but it will all be related to the articles, including our presentations on sample size calculation, some related statistics in inferential statistics portion, and the sessions outside. So there's been a little slight change in program requested by the hosts at Comstech. And so after Dr. Abdul Rashid from Drug Regulatory Authority of Pakistan talks about the current clinical trial scenario in Comstech region primarily, but he may allude to other areas of South Asia as well. Then we are going to have the certification ceremony immediately after that at 11:40, inshallah. So the session that Ms. Khalud Janjua from Turkey is going to be conducting on sample size calculation will be from 12 o'clock to 12:40, inshallah. And from 12:40 to 1:20, Ms. Maham Zahid is going to be doing a session on inferential statistics. After that, we will still have 70 minutes, 1 hour, 10 minutes for the Jummah prayer and lunch. So most places out here, those of you who are from local areas, should be able to get your Jummah prayers and lunch in that time slot. And we will reconvene, inshallah, at 2:30. Most of the prayers in this area would be around 1:50 or 2 o'clock in any case. So we should be all back by 2:30 safely, inshallah, and be back on our schedule. So the certification ceremony at the end will obviously be done earlier. So we will probably be able to close we probably be able to complete our sessions a little ahead of time, and we may be able to take some extra questions if you feel there's unanswered questions. So Dr. Abdul Rashid, I guess, is here on site, but his presentation, if it's been uploaded, if you can request him to kindly come in. So before that, I just want to tell you that the sequence of events today, after the sample size calculation and the inferential statistics, then after Jummah from 2:30 to 3:10, I'll try and put together things that we have done since yesterday by going through the elements of the consort statement. So hopefully in those 30, 40 minutes, it will be a review of all those activities. And then we will have, of course, the critical appraisal and the proposal writing session. So Dr. Abdul Rashid is here. So you just give me the signal and we will start then. Okay. Yeah. So in the afternoon, after we're done with the consort statement, then we will, inshallah, go on to critical appraisal of randomized controlled trials. And I was mentioning yesterday that Professor Mubin Iqbal is one of the pioneers of evidence-based healthcare and educationist, academician, researcher, as well as a very, very, very well recognized pulmonary and critical care specialist. So he's one of those people who's taken the holistic approach to academia very seriously. Again, a rare breed in Pakistan. And so he will, inshallah, take you through the critical appraisal of at least 1 or 2 of these articles that were also distributed. And then you will, inshallah, at the end, we'll close down. If there are any things that need to be clarified before Dr. Abdul Rashid, whenever you're done, you let me know, sir. So when there are any things that you want to clarify from yesterday, but before that, I promise that I will return some fulus, some money to the people sitting out here. And what I will do is I will hand over this amount to Mr. Mufassir again. Thank you, sir. And whoever could not answer yesterday, it was all part of good faith exercise. So we want to make sure you get your money back. So if you can just raise your hand and how much money you lost yesterday. You're not gambling. It was just a little bit of an incentive and it was just a little bit of a game. So then Mr. Mufassir will come back and return that money to you. So thank you for being a sport yesterday. Normally, I usually only take half the money for not answering correctly. And usually I make up because lots of people don't answer. So even that money actually comes back to me. So at this point, if there are any questions or clarifications from yesterday, Dr. Abdul Rashid's presentation is ready. Okay, all set. You have something done? So
Okay.
if you can just put it up here, and we'll get started.
मेरे ईमेल पे नहीं आया कजीमा को भेज दी थी RSHI लिखें RASHI जी
Sahir. Sahir, I have shared Dr. Saab's presentation on the Zoom as well.
Sahir, please mic unmute करें.
It's okay. Yeah. Assalamualaikum everybody and good morning. If the time will be different in different OIC countries and other parts of the world, so good morning, good afternoon, and good evening if there is any time difference. So this is this is a map of the OIC member countries, and पिछले नहीं as I mentioned, because the device was not coming, the scenario of clinical trials among OIC member countries. This is the map of the 57 member countries throughout the world, including Pakistan. This is map of the Islamic Republic of Pakistan, having 4 provinces, ICT, AJK, and DB. This is one of the lush green area of Pakistan, Islamabad, the capital of Pakistan, very beautiful city. And Islamabad is the #1 beautiful city of the world. A beautiful capital of the world, not city. Islamabad is the #1 capital, beautiful capital of the world. So this is one of the picture. Another one, and also the one of the third biggest mosque, that is Faisal Mosque. Very lush green area of Islamabad. This is the demography of Pakistan. You can see the population, 220 million population. Yesterday I met with the ambassador of Denmark. She was saying there 6 million. I say 6 million is one city of Pakistan. So we are sixth and hoping to be a fifth populous country of the world very soon. This is the Programmability Authority of Pakistan. It has been promulgated as a form of DRAP Act 2012. It is very latest, not just 9 years before. And Alhamdulillah, I was a technical person who made this law. And first time in history of Pakistan, after the Drug Act in 1976, I made the changes, and first time I included the herbal, homeopathic, nutraceuticals, medical devices, blood and blood products, traditional medicines, Unani medicines, dairy and milk and dairy products. Before there was no law in Pakistan, but I included in DRAP Act 2012. The head of institution of DRAP is Chief Executive Officer, and we have 13 different divisions. I am one of the director of Division of Pharmacy Services. I have heard that there are changes. They want also they are bringing changes in the DRAP Act and making a 14th division as a research and development division, but it is not still implemented, not approved. इसकी सेटिंग जरा ठीक कर दें. ये जो उसकी आ रहे हैं. Clinical trials comes under the control of Division of Pharmacy Services, and I am head of that division, also the chairman of the Clinical Study Committee. We have the rules, that is Buyout Study Rules 2018, and we work according to these rules, and Clinical Study Committee is working under this rule. In these rules, we have to we deal with clinical trials, research and development, advertisement and ethical marketing, and also the pharmacovigilance. Also, we deal with antimicrobial resistance, and clinical trials are one of our responsibilities. As I mentioned, that DRAP Act gives the legal and mandate to the Division of Pharmacy Services, and preamble of this is that biostudy rules apply to all contract research organizations, that is CROs, laboratories for clinical research, bioavailability and bioequivalence studies center, and organizations operating in public and private sector involved in clinical trials of therapeutic goods. This is the composition of the Clinical Study Committee. One clinical pharmacist, one professor of pharmacology, one professor of pharmacy, one clinicians, one statisticians, one representative from Pakistan Pharmaceutical Manufacturer Association, also the one representative or observer from Pharma Bureau. We have opted 10 members of different specialties to utilize their expertise in different fields, either it is medical devices, either it is the biological vaccine and sera, or the allopathic drugs, or the animal medicines, or ventilator, anyone we can swap them. And the clinical study committee comprises of the Vice Chancellor, which is very prominent in Pakistan, is very famous physician. He is the Vice Chancellor of University of Health Sciences, the Professor Dr. Javed Akram, also the professor from Faculty of Pharmacy, also the retired brigadier, and one of the very famous statistician from Shaukat Khanam Hospital, Lahore. All the vaccine during COVID, we all remain in contact, and at national level, we have National Vaccine Committee. I'm also co-opted member of that vaccine committee. So before coming to the draft, the COVID vaccines are first, they are approved from National Vaccine Committee, then the application for emergency use authorization comes to draft for the approval and for availability of COVID vaccine in Pakistan. Roles and responsibilities of the Clinical Study Committee. On behalf of my division work for screening, assessment, review, and evaluation of applications for clinical trials, bioavailability studies, and also the CROs, screening, assessment, review, and evaluation of applications for the session of clinical trials conducted, and also for bioavailability and for bioavailability studies, inspections of the premises where clinical trials have to be conducted is mandatory, and we go 5 people. One is clinicians, one is from basic sciences, another is from biostatistician experience, fourth one is the pharmacy expert, and the fifth one, either I, as a coordinator from lab, I goes on every clinical trial site for physical verification and to observe whether they meet the international standards of clinical trials or not. We 5 people go and verify the facilities. Also, if we grant, reject, or suspend license for clinical trials, depending upon the inspection report. Rules give us authority to either grant, reject, or suspend the clinical trials during 1 year period if they are not meeting the requirements of the clinical study, which we have mentioned in our rules, are either decided by the clinical study committee. We can suspend the clinical trials, and we can also conduct inspection and see the progress report. Interim progress report, final progress report is submitted to clinical studies, and we assess either they are going according to protocols or not. I want to mention one thing, that every clinical site, they have to take approval of the study from their institutional review board, that is IRB, and also the National Bioethics Committee. If the application does not complete these 2 requirements, we did not consider their application for clinical study committee. So it is mandatory that IRB and National Bioethics approval must be there. When we started this clinical trials in 2018, after the promulgation of DAP Act 2012. And the first clinical study committee was conducted in February 2019. In July 2019, we have given the first clinical trial site of Holy Family Hospital, Rawalpindi. That was the international multi-country trials, that is Women 2 trial for tranexamic acid. So this was the multi-country trials, and more than 6,000 subjects, they have gone to the trial. In July 2019, we gave the first CRO, that is DRK Pharma Solutions, and in November 2020, that we give the bioanalytical lab to Pakistan Drug Testing Research Center, Lahore. Also, the first bioequivalence centers, that is pharma professional solutions, are also the where is Professor Raza Shah? Is that was ICCBS, HEJ, University of Karachi. That was the second bioequivalence centers in Pakistan. The first study which was approved in May 2020, that was the first bioequivalence studies. And the first vaccine clinical trial in Pakistan, that was the Sinopharm, which is widely used in Pakistan now. It was a great risk for me and for the committee, because before that, none of the clinical trial on vaccine was conducted in Pakistan, particularly phase 1, because it was a great risk. Alhamdulillah, that has been succeeded. Indus Hospital and ICCBS, they conducted jointly. In June 2020, we have conducted the phase 3 vaccine clinical trials, that is CanSino, biologicals, adenovirus type 5 vector COVID-19 vaccine. It was approved, and you will be happy to listen that this CanSino vaccine is being manufactured in NIH Islamabad, and the bulk comes from China, and the filling totally it is manufactured in NIH, not bulk, but after refilling. But the raw material comes from China and manufacturing is done in NIH, because this I have clarified from the chief biological NIH. The first medical devices, that is a capsule endo microscopy, was approved in July 2020. You will be astonished that we have also approved the first traditional Chinese medicines, that was also the Jinhuang granules for treatment of COVID-19. It is also given to ICCBS, HEJ Karachi University, by the Professor Raza Shah Sahab and Nagma, maybe she is here also. And as a regulatory body, in short period of time during COVID, because it was very difficult for us to inspect the premises, we always say that please don't bring us near to COVID ward, but they say this is the COVID ward. So it was very difficult for us to conduct inspections. Up till today, we have conducted in 1.5 years, 1.5 years, we conducted 31 clinical study committees. In 2 years and 7 months during pandemic, and up till now, as previously yesterday I mentioned that we have approved 75 clinical trials, 35 clinical studies in Punjab, Khyber Pakhtunkhwa, Sindh and Balochistan, and Islamabad capital. It means we have 4 provinces and ICT. We have approved clinical trials and studies all over Pakistan, covering all the area of Pakistan. Totally 75 clinical sites and 35 studies. These studies are either multinational or the national, all included. We have approved 10, up till now we have approved 10 contract research organizations, 2 bioanalytical labs, and 4 bioequivalence centers have been given the license. 5th, we have conducted inspections and it has been recommended, but will be approved in next meeting of the clinical studies committee. This is the current situation of clinical trials of different phases in different Muslim countries. I cannot mention all the one, but I have taken some countries, that is Jordan, Bangladesh, Indonesia, Saudi Arabia, Pakistan, Iran, Malaysia, Egypt, Turkey, and the source is the US clinical trials GOV. Maybe it is correct, maybe there will be some studies will be approved from their regulatory authorities, but there will be some studies they are not approved from their own regulatory authorities. But this is the whole data of some Muslim countries where you can see that clinical trials are going to be conducted and in different phases, other phase 0, phase 1, phase 2, phase 3, even the social studies, they are also included in that. All the guidelines, this is achievement of our division, that all the guidelines are available in dra.gov, their websites. All the stakeholder applicants and general public, they can see what are our guidelines. All the things are available online, you can see. And also, we have prepared a software from our own division, that division of MIS, and all applicants, they are bound to apply online on this site, DRIS.dra.gov.pk. So now it is mandatory that all the applicants, they must apply through online. Hard paper is not acceptable. So recently we have done this one, and our division of pharmacy service have prepared this software. User manual is also used in from 24th August 21 for any news, any alert, so that anybody can access our all information. All the rules, regulations, and fee schedule are available on DRAP official website. So there is no need to conduct on telephone or fax or any other conditions. All the things are available, law, rules, regulation, SOPs, application forms are available online. So this is a great facilities. Moreover, Drug Regulatory Authority of Pakistan has adopted ICH guidelines, and also we have adopted US clinical trials.gov. Any applicant, after getting approval from DAP, they can upload their application, our approval at the clinical.gov US FDA website page. They can upload this. So their all information can be, whole world can approach the approval of from DAP on US clinical trials.gov. These are the different multi-country trials. If USA and Brazil is there, Pakistan is also there. Another study is USA, Australia, Germany, Netherlands, Spain, United Kingdom, and Pakistan. So along with the European countries, Pakistan is also there. And another study is from Pakistan, Australia, and New Zealand. If Deliver is another study, this is multinational, multi-country trials. USA, Belgium, France, Canada, Bulgaria, Germany, Greek, many countries. So if phase 3 is at serial number 8, if there are 26 countries, then in phase 3, Pakistan is the 27th country. So this is our great achievement. I want to say that if clinical trial is going to be conducted in developed countries, Pakistan is also one part of them. If clinical trials are conducted at developed and underdeveloped country, Pakistan is also one of the center where that clinical trial is going to be conducted. This is corona vaccines studies, and you can see that clinical trials are conducted in different diseases. You can see a huge list, up to 15 different on different diseases. The clinical trials are conducted. You can see at the postpartum bleeding in women, and a long list. I cannot. Public access, all the sites, studies, they're also available, as I mentioned, in the on the website. Also, the minutes of the committee, that is clinical study committees, are also available on website of the track. As I've already mentioned, that international access is also there. Any country can access to the approved sites of the Pakistan and approved studies. This is international. And what is the way forward? That's I want to suggest one thing. After coming to His Excellency Professor Dr. Chaudhry Mohammad Iqbal, he is a vibrant personality. we want to take benefit from his, as a coordinator general. We want that there should be giant research and development fund shall be created in OIC member countries, and it giant fund shall be established as OIC has recommended during their meetings in 2016. They have already recommended, they have already approved that all 57 member countries should have a giant research and development fund, and we request to the Chaudhry Iqbal Sahab that practically work should be done, because now 5 years has been passed. This is also, I will request to all the participants that we should make the recommendation of this 2 days workshop that giant R&D fund should be established with the support of Islamic Development Bank. Collaborative clinical studies should be conducted among member countries, that is 57 countries, as per Islamic point of view. In some cases, we are interested to conduct clinical research in fasting and non-fasting, and other रोजे की हालत में या रोजे के बगैर जो है हम चाहते हैं कि ऐसा, या जो इस्लामिक कंट्री में जैसे हमारे 15 शबान का रोजा है, हम उनको जो है हम कंपैरेटिव स्टडी करना चाहते हैं। इस्लामिक पॉइंट ऑफ व्यू से हम चाहते हैं कि क्लिनिकल स्टडीज हों वो इस्लामिक कंट्रीज में। Also we want to conduct the research work either for treatment of the COVID, not vaccines, but also the oral tablets. This should be jointly clinical study should be conducted either at 57 national countries at the regional level or at national level, but we should conduct the clinical trials on COVID-19. A meeting should be conducted in between 57 Muslim regulatory authorities countries. This is very, very important, but we need the support of the comstech. If Chaudhry Iqbal is here, their officers should convey to them that we want that 57 regulatory authority countries of the Muslim community, they should jointly conduct a meeting so that we can go ahead. Also, this is our one of the recommendation that jointly of OIC member countries should conduct conferences, workshops, seminars. Either they should be hybrid, that is physically or virtually, should be conducted not only on vaccines, and medicines, allopathic medicines, but also on herbal and traditional medicines, and also the medical devices, because this is the need of the 57 Muslim countries. Animal studies and preclinical should be conducted. Phase 0, 1, 2, 3, and 4 should be phase 4 clinical studies should be conducted jointly. Academia, research and development institutions, medicine regulatory authority shall coordinate with each other and make focal points for joint clinical trials. This is very, very important because you cannot contact different institutions. If there is one focal point in one country, so we can make coordination and communication and liaison with each other. So this is my suggestion, that from comstech, there should be some focal points, their email address, their office phone numbers, their mobiles, all the communication media. We should also be connected with either by the social media also. With this, thank you very much. These are my suggestions. Also, the current situation of clinical trials in Pakistan, also the current situation of some of the member countries of Islamic 57 countries, and also the recommendations from Pakistan from this 2 days workshop. Other recommendations should also be included, so that these recommendations should be placed before the executive committee board of the OIC member countries through Professor Dr. Chaudhry Mohammad Iqbal, Coordinator General. Thank you very much. If there is any question, please welcome.
Thank you, Dr. Abdurrashid Saab, for going through the on the ground situation in terms of regulatory aspects, in terms of the infrastructure, in terms of the processes that have been put together very diligently over the last, at least since the DRAP Act of 2012 and the 2018 biostudy rules. A lot of activity has happened in the last 1.5 years. I think one needs to commend and recognize that. And that's what I think we're all here to build upon, inshallah, together, so that we can utilize some of these, the infrastructure locally, combine it with capacity building exercises, join hands, inshallah, amongst the OIC comstech purview, and hopefully take all this effort forward. I thank you, sir, for a very comprehensive presentation. And moving right along, we are going to invite Fulu Janjua from Turkey to come online and unmute herself and get online. Can I get some assistance from you, please? And so we'll do that. And then subsequently, the Coordinator General, Iqbal Chaudhry Saab, is going to be coming in and distributing certificates. And then we'll go along with the next presentation, which will be on inferential statistics. So Ms. Fulu Janjua, I just want to introduce her. Dr. Fulu Janjua now obviously has been in the field of clinical trials for the last 7 or 8 years that I know of. She's a PharmD with a MS in biomedical sciences and as a PhD scholar, completing her PhD in biomedical sciences. She's also a SOCRA approved clinical certified clinical research professional. And she worked very closely with my team, both at Shifa International Hospital when I used to work there, and then subsequently out at Maruf International Hospital. Now she's gone back to complete her PhD work. And she's also a diplomate of the American Board of Quality Assurance Utilization Review. So putting all that together, putting in the quality assurance processes, she was one of the instrumental persons who helped put us together the infrastructure that was subsequently approved by Drug Regulatory Authority of Pakistan as a clinical trial site. So I need to acknowledge that, and I thank her for that. And inshallah, you'll enjoy our presentation, hopefully. Fulu, there, you can hear us?
Yes, sir, I can very well hear you.
Thank you. All the best.
Thank you so much, sir, for such a nice introduction. Good morning, good afternoon, and assalamu alaikum to everyone. Bismillahirrahmanirrahim. My presentation today is about sample size calculation, and I would take you through all the different steps that you have to follow when you first start your study, putting a particular focus on randomized controlled trials. So one of the questions that you come across whenever you're starting your study is, whether it's an observational study or intervention study, is what is the number of sample size that you should be including in your study. So we're going to go through the practical aspects, how you take different parameters, ones that you decide and ones you take from your existing studies, and how you put them into a calculator to find you a number that is reasonable enough to detect a difference if it exists, and gives your study the power enough to detect a difference of significance. Please feel free to post your comments in the chat section, and we'll have a 10 minutes question answer session after the end of this presentation. So before starting your research, some of the questions that you come across is, which participants should be included? How many participants do I need to include? What data needs to be collected? And how will I analyze my data? Now, keep in mind, all of these questions, they need to be addressed at the time when you're writing down your proposal, or you're applying for a grant, or you're applying for an IRB or ethics committee approval. These are not questions that can be left up till you have collected your data and you're thinking of analyzing your data. These are the questions that need to be addressed a priori before starting your study. Now, why do we care about sample size? This is a question that I've come across multiple times when I was calculating sample size for different studies or assisting people in their research, or also when I was doing the statistical analysis for different studies. This is something that we come across very often. There have been times where people have done their work very well. They have a very solid study question. They have a well designed research protocol. They have done their due diligence by collecting their data. But when it comes to the analysis, it seems deficient because they did not really consider the sample size calculation. And now the study is either underpowered or they cannot justify that number of subjects that you took to either the journal that they're submitting to or their grant committee. So why is it important to consider sample size? Some of the options that are available that some of you might have heard of, why not enroll all the subjects available? So let's suppose you're working at a facility, you're working at a hospital or a clinic, and you're trying to conduct a study, and you're trying to enroll some people with certain diseases, and you have certain subjects at hand. Let's say your clinic sees about 2 subjects per day, 2 patients, eligible patients per day. So 2 into 5, 10 per week, and 40 per month. So if your study goes up to 6 months, so why not take 240 subjects? Or maybe set a time limit. You take all the participants visiting during 6 months. So let's say you have starting from now up until 6 months or 1 year to analyze your data and write up a research paper. So why not take all the subjects that are coming in during that time period? Now the question arises, what if there are too few subjects that come to your facility during those 6 months? What will happen if we enroll too few subjects? And the opposite can also happen. What if you have too many people coming in? Maybe you work at a very busy tertiary care hospital and you see 1,000 subjects that fulfill your eligibility criteria. So what will happen if we enroll too many subjects? And then there's an approach that certain new novice researcher take, is that I can start the data collection and bother about statistics later. Now, something that I have emphasized before, that this cannot be done. Why? When you enroll too few subjects, your study does not have the power to detect the difference if it exists. So even if you're comparing in an RCT, your interventional drug with a placebo or a control, even if your interventional drug does a lot better than the placebo or the control drug, you might not be able to show that difference. Why? Because the sample size was too small. Your p-value might not be significant, okay? And then the other question, what will happen if we enroll too many subjects? So starting off right from off the bat, too many subject means a lot of resources. Too many subject mean that you have to allocate a lot of people, human resource, a lot of time, a lot of energy, and a lot of money to it. And there's also an ethical aspect to it as well. So as Dr. Shumail might have walked you through all the ethical aspects that you are concerned with whenever you are conducting an RCT, you cannot expose more than a minimum number of subjects to risk without any explanation, a reason for it. So let's say your sample size comes out to be, if you're calculated, your sample size comes out to be 200. Let's say we have also included the dropout rate, and we have also considered the iteration rate. You cannot simply enroll 400 or 600 subjects. Why? Because the more subjects that you take, you are actually exposing more of them to a risk. And what is the risk? Whenever you're conducting an RCT, whenever you're conducting an intervention study, there's always a risk associated with the treatment that you're giving your interventional group. So there's also an ethical aspect to it. So you cannot enroll too many, and you cannot enroll too few. Now, in studies that are concerned with detecting an effect, so by that we mean that you are comparing an active group and a control group. So, and you're trying to determine that what is the difference between the effect. If an effect deemed to be clinically or biologically important exists, then there is a high chance of it being detected. That is, that the analysis will be statistically significant, and the p-value that you get if the effect actually exists, if your drug is doing better than the control or placebo group, then there are higher chances, much higher chances, that the p-value that you get would be less than 0.05, which is the alpha value that you set. And if the sample size is too small, then even if the large differences are observed, it will be impossible to show that these are due to anything more than sampling variation. So the p-value would be more than 0.05 in that case. And because the inter-individual variability would be too much for the group to be compared in a logical basis. So when you start off with sample size calculation, you might be overwhelmed by the terminology that you come across. I'll try to walk you through one of what these terminologies means and what are the values that you set and where do these values come from. So some of the terminologies that you will come across is a significance level. This is the cutoff point for the p-value below which the null hypothesis will be rejected, and it will be concluded that there is evidence of an effect. This is typically set at 5%, or if you take it in numbers, that is 0.05. This could also be 2.5% if you want your study to be one-tailed, your question to be one-tailed, one-sided. And if you want to be more conservative in some studies, this might be set at 1% or 0.01. But bear in mind, as you reduce the value, it becomes more and more difficult to prove a difference, right? But on the other hand, it gives you more surety that yes, the results that you are coming across, they have a very few chances of making a false positive error that we're going to discuss in the future slides. Now, whether your study is one-sided or two-sided tests should be used unless there is a very good reason for doing otherwise. So what a two-sided test means that when you're comparing drug A versus drug B, there's an equal chance that the drug A would do better than drug B, or the drug B would do better than drug A. And let's say you are comparing your interventional drug, a new drug with an old one, or a new drug with a placebo, the same goes. But if you have a very good reason to consider that no, the drug that we're testing would definitely do better than a placebo, then you should go for a one-sided test. Again, if you go for a one-sided test, the significance level becomes half, becomes 2.5 or 0.025, and it becomes twice as much harder to prove the significance. Then power is very important. This is something that I have mentioned in my previous slides. Your study is underpowered or adequately powered. What does it mean? Power is a probability that the null hypothesis will be correctly rejected. That is, rejected when there is indeed a real difference or association. It can also be thought of as 100 minus the percentage chance of missing a real effect, which is actually the false negative error, or the beta. Therefore, the higher the power, the lower the chances of missing a real effect. So this is something that's inversely proportional. Power with beta. Power is typically set at 80%. You cannot go below 80 because then that's not A, not academically acceptable, and B, the chances of committing a false negative error would be just too high. You can also set it 90% or 95% or 99%, but not below 80%. So just to reiterate, alpha cannot be more than 5%. The significance level cannot be more than 5%. And the power cannot be set less than 80%. That's the threshold that the scientific community has agreed upon. And now, something that is very particularly important in case of RCTs, the randomized controlled trials, the effect size of clinical importance. Now, it's not just important that the results that you get are statistically significant, that is, the p-value comes out to be less than 0.05. It is also important that the effect size that you're measuring, that is of clinical importance as well. So what is effect size? It is the smallest difference between the group means or proportions. Means if you're comparing two quantitative variables between group A and group B, your intervention and control, or proportions, that is the percentages, if you are comparing the odds ratio or the relative risks. And this is the difference that would be considered to be clinically or biologically important. Now, this difference comes from the researcher, comes from the clinician. If you're not too sure, if you're somebody who belongs to basic sciences and you're collaborating with a PI or you're collaborating with a physician who is actually an expert of that field, then you might ask them, what is the difference that is important to the clinician or to the patient? So you cannot go for something very, very minuscule just to prove that there's the difference actually exists. That difference has to matter to the patient, to the subject, or to the medical, scientific, clinical community. The sample size should be set so that if such a difference exists, then it is very likely that a statistically significant result would be obtained. Now, at this point, I would like to mention that the greater this difference is, difference between group A and group B, your intervention and your control, then you would need a smaller sample size to prove that difference. But if the difference is too small, even if it is clinically significant, then the sample size needed would be large. So it is inversely related. So just to go through some of the aspects the basic principles whenever you conduct a statistical inference is that you draw a sample from the population of interest, okay? You analyze that same sample data and you make conclusion about the population based on the results from the sample. So you're not studying the whole population, you're taking a small subset of the population, but whatever results that you get from their study, you're trying to generalize it. So whenever you conduct statistical analysis and you infer a result, are always correct and incorrect conclusion. So we draw a 2x2 table, which helps it understand what the false positive and the false negative errors that I was talking about. So on this side, we have the conclusion that is based on the sample, meaning that this is whatever results you are getting after you have conducted your study and you have conducted the statistical analysis. On the top, you have the true state, which is unknown. This can only be known if you have studied each and every member of the population, which is obviously not possible, right? And then we have divided into four sections. When the true state shows that there is no difference, that is the null hypothesis is a true, or the true state could be that there is a difference. The null hypothesis is to be rejected, and there actually exists a difference between the placebo or the control and the intervention group. And here you have two possibilities based off of your conclusion. You reject the null hypothesis, that is, you have gotten an evidence that their difference exists, meaning that the p-value was less than 0.05, or you fail to reject null hypothesis, you did not get any evidence of difference, that is, the p-value did not come out to be less than or equal to 0.05, it was more than 0.05. So what are the different conclusions that you get? So, if the true state shows you that there was no difference, actually your intervention, the drug did not do better than the placebo, but your conclusion shows you that there was a difference. Then you committed a type 1 error, which is false positive. Your results showed you a positive result, but actually it's false, so it's a false positive result. Then you have two correct conclusions where there was a difference and your study showed you a difference, then that's the correct conclusion, which is true positive. The other true correct conclusion that you can come across is that there was no difference actually, and your study showed you the same, that there was no difference. So you got a correct conclusion that is true negative. Now, the last one is where the difference existed. The drug did better than the placebo, the intervention did better than the control, but your study failed to show you. So this is a false negative error. It is very important that you learn the difference between these two, and you also learn the terminologies associated with it. So just to review, type 1 error is false positive, type 2 error is false negative. So what is alpha and beta that we talk about and that you might come across whenever you are calculating your sample size? So alpha is a probability of making a type 1 error, and that's closely related with your significance level. So if I say that I'm setting my alpha as 5%, then I'm saying that I'm giving myself a margin of 5% for probability of making a type 1 error, or a 2.5% probability of making a type 1 error. And beta is the probability of making a type 2 error. So like we learned before, that your power is 100 minus beta, or your beta is 100 minus power. So if your study had a power of 90%, that means there is a 10% probability that you might make a type 2 error, which is a false negative error. Now, bear in mind that as much as we might try that our study results should be as accurate as possible, it is impossible to bring these probabilities down to zero. There's always going to be a probability that you're making either a type 1 error or a type 2 error. Why? Because we have not studied the whole population and we're only basing our results off of the sample that we took. And there's always a trade-off, meaning that whenever you try to reduce one, you might have to increase some other value, or if you decrease one value, you might have to trade off some other value. So just to learn this, your power and your beta are inversely proportional, right? So if you keep the sample size same and you increase the beta, that is the probability of making a type 2 error, then your power comes down. Okay, so similarly, if the power and the sample size are also inversely proportional, sorry, the power and the sample size are also related in a way that if you increase your power, your sample size goes up as well. And if you keep both of these same, alpha and beta, they are also inversely proportional. So we would test our values and try to see the conclusion that we come up with whenever we're calculating the sample size. Now, lastly, what is the connection between alpha and p-value? So we talked about alpha, which is the probability of making type 1 error. If the p-value is less than alpha, then a hypothesis is rejected and the result is declared to be statistically significant at the 5% value, or the 2.5 if you're going one-sided alpha. And if the p-value is greater than alpha, the result is not statistically significant at the 5% alpha level, and it means that you have failed to reject the null hypothesis. Some of the sample size calculators that you can use that are easily available and accessible are the WHO sample size calculator, the OpenEpi that the link I've provided here, and the MedCalc. There are many sample size calculator, and all of these use the same values. They might be mentioning the terminology in a different way. For example, one sample size calculator might ask you to put in the value of beta, while the other might ask you to put in the value of power. One might ask you to put in significance level, and the other might ask you to put in the alpha. So I hope I have made the difference clear. And how do you calculate the power if you have the value of beta? Now, things to know before calculating a sample size. You must know the value of alpha or significance level, because these are both related. You must know the value of beta or the power, and you must know the values obtained from previous studies. So whenever you're doing your literature search, we highly recommend that you conduct a thorough literature search and you come up with a study that is closely related to the research study that you are about to conduct. And the population is also the population, and the study question is closely related to what you are intending to study. Now, there's a situation arises when you are trying to study a newer drug and there's no previous evidence on that. There's no phase 1, there's no phase 2, has not been conducted in any other country. In that case, you might go towards conducting a pilot study. You might take 50 subjects, you might compare your intervention and your control, and you might use those values to put into the sample size calculator. Otherwise, you do a thorough literature search and you pick up the values from the studies closely related to the one that you're about to conduct. Now, whenever you're studying the previous literature, which variables should be included in the sample size calculation? The sample size calculation should relate to the study's primary outcome variable, right? So the primary outcome that you're studying. But if the study has other secondary outcome variables, which is also considered to be important, as this is often the case, then the sample size should be sufficient to do the analysis of these variables as well. So let's suppose that you have one primary outcome variable, and then you have 4 other secondary. So you might get 5 values of sample size, but you would choose the one that is highest. Why? Because the highest value of sample size would ensure that your study is powered enough to detect all the primary and the secondary outcome variables. The value should also be consistent with the study's aim and statistical analysis. If the aim is to demonstrate that a new drug is superior to an existing one, then it is important that the sample size is sufficient to detect a clinically important difference between the 2 treatments. The sample size calculation should also be consistent with the study's proposed method of analysis, since both the sample size and the analysis depend on the design of the study. Is it going to be superiority trial, non-inferiority trial, or an equivalence trial? Now, whenever you're comparing comparative studies, you might come across either comparison between means, then you'd be using quantitative variables. Then you would be asked to use the mean and the standard deviation reported in the previous studies. You can take one expected value and use the other as anticipated value. Or as it is usually done in RCTs or randomized controlled trials, you use an effect size, which is the difference between the outcome of the 2 interventions. It could be 20%, a small, medium, or a large one, a 20%, or 40%, or 80%. So what you're trying to show is that your intervention works 40% better than your control group. Now, again, this clinical significance is to be determined by the researcher. whether they think that 20% is enough or they think that 80% is something that is meaningful to the clinicians or to the patients. And you should also account for the dropout rate. If you have anticipated that the dropout rate is 10%, or if the study design is too complex and the compliance is low and you are expecting the dropout to be 20%, then you should add that to your final sample size so that your study's power does not go down when you consider all the dropout rates. So now we'll move towards one of the examples that we studied in. We provided you with the research articles when we shared the papers with you. So I'm going to walk you through the examples of two research articles and try to show you how they use the sample size. So one thing that you have to bear in mind that whenever you are applying for an ethics committee approval, you have to justify your sample size. But in some case, if you did get an approval and no one asked you for the sample size, now when you are submitting your manuscript for the publication in a journal, every journal would ask you to justify your sample size. So let's take the example of the recovery trial, where they used dexamethasone in hospitalized patients with COVID-19. So this is a screenshot that I took from the actual article. Now we're going to do some reverse engineering and taking these values, I'll put it into the sample size calculator and show you how they come up with the number that they used. So the question in this recovery trial was, does dexamethasone improve 28 days all-cause mortality in COVID-19 patients compared with placebo groups? Okay. Excuse me. So this is the part in the article where they show you the statistical analysis, and I took a screenshot. So what they did was that as the trial progressed, the steering committee, they took the numbers that were given in the outcome effect, and then they used the trial because, like I discussed before, COVID-19 was a new disease, and there did not exist sufficient evidence before to use in the sample size calculation. So what they did was that they did not calculate the sample size when the study started. Instead, they started the study and then estimated the numbers. So what they determined was if 28-day mortality was 20%, then they enrolled 2,000 in dexamethasone group and 4,000 in the care group. How? They took a power of 90%, a two-sided p-value of 0.01%. So if you recall, we said that the p-value could be less than 0.05. And if you want to make it more conservative, if you want to be more sure, if you want to reduce the chances of the false positive error, then you can go up to 0.01 or even lower than that. But this makes it more difficult to show the statistical significance. But it gives you more surety that the drug actually worked. So the outcome effect they're looking for is the clinically relevant proportional reduction of 20%, an absolute difference of 4 percentage points between the two groups. So all-cause mortality, what they're looking for is they're looking for a difference of 4 percentage points. So how does this all come in? When you open your sample size calculator, in this case, I've used the value in Open Epi calculator. These are the variables that you're asked to put: the power, the alpha, the proportion of subjects with outcome of interest in the placebo group, which in this case was all-cause mortality, the anticipated proportional reduction in the outcome of interest, how much reduction in all-cause mortality are we expecting in the dexamethasone groups. Now, the final anticipated proportion of subjects with outcome of interest in intervention group, and the ratio of placebo to intervention group. Now, I would like you to open the manuscript itself and try to fill in the values yourself when you are calculating it. So the power the researchers took was 90%. This comes from the article. The value of alpha is 0.01 or 1%. The mortality in the placebo group was 20% or 0.02. The reduction that they're looking for is 20% of 20%. So here is the calculation. 20% of 20% is 4%, and 20 minus 4 is 16%. So now we're anticipating that the placebo group would have a mortality of 20%, and the dexamethasone would have a mortality of less than 4 percentage point less, that is 16%. And the ratio of placebo to intervention group is 2 that we read in the article. So now this is what we come up with. alpha, the two-sided significance level is 1 minus alpha, which is 99, 90% power, ratio is 2, percentage of unexposed, placebo with outcome is 20, exposed with outcome is 16, and these are the numbers that the sample size calculator gives you. The sample size of exposed is 2,000, non-exposed is 4,000, placebo and intervention, and the total sample size is 6,122, which is very close to what the study itself had quoted. So now you can see how the RC, whenever you're conducting an RCT, how the major, the research group, they have calculated their sample size. Moving towards the other example, the CKD trial, to assess the long-term efficacy and safety of the CGLT2 inhibitor in patients with chronic kidney disease with or without type 2 diabetes. So again, I'm taking a section from the manuscript where they discuss the parameters that they took for sample size calculation. So the sample size, the outcome events was 22%. They estimated that 681 primary outcome events would be needed to detect a 22% lower relative risk in the dapagliflozin group than in the placebo group, a hazard ratio of 0.78 with 90% power using a two-sided alpha level of 0.05 and assuming an annual event rate for the primary outcome of 7.5 in the placebo group. So the primary outcome that they're looking for is 7.5 in the placebo group. And what they're hoping for is a 22% reduction in the SGLT2 group. And type 1 error at a two-sided 0.05 level. So again, the variables needed for the sample size calculation are the same: the power, the alpha, the proportion of subjects with outcome of interest in placebo group, and also the intervention group, and the ratio of placebo to intervention, which was 1 in this case. 90% power, this comes from the study. And alpha was set as 0.05. The proportion of subjects in with outcome of interest in placebo groups, that is CKD, in the group that received placebo was 15%. How did we come to this value? It was an annual rate of 7.5, and the study was conducted over 2 years. So 7.5 into 2 becomes 15%. So the placebo group basically experienced 15% outcome of interest within 2 years. Now, what are we anticipating? We're anticipating a reduction of 22%. So 15% into 22 is 3.3, 15 minus 3.3, 11.7. So what we're anticipating is that the intervention group would only have the outcome of interest, the events of 11.7%, and the ratio of placebo to intervention is 1. Now, when you factor in all these variables, this is actually the result that you get from the open epi sample size calculator. The two-sided level of significance, 1 minus alpha, 1 minus 5 is 95. The power is 90. The placebo and the intervention group, the ratio is 1. Percentage of unexposed with outcome is 15. Percentage of exposed, that is intervention with the outcome, is 11.7. And this is the sample size that you get. 2214, and the total sample size is 4400. And this is close to the sample size that they took in this study. So we have we had enough time to only discuss 2 examples. You can go back and look into the articles that we provided you and open either WHO sample size calculator or the OpenEpi or the MedCalc and try to practice this. And whenever you are about to start your own study, I would advise you to look at these, set the level of alpha, set the level of beta, and try to look into the clinical, if the effect size that you're trying to look into, and then calculate your sample size before you have started your study. This is the end of my presentation. Please feel free to give your questions or your comments. The floor is yours.
Thank you very much, Khulud. Again, she has finished in great time with great time management, and we have 10 minutes now to clarify your concepts. I'm sure this is not a statistics workshop, and you need a lot of mentoring. You're not all expected to be biostatisticians. Khulud is not a biostatistician either, but as a pharmacist who has been involved in clinical trials, she makes it a point to understand what she's doing. So as a clinician and as a clinician researcher, it's important for us to understand the implications of sample size calculation. The biostatistician is not going to do something from his own, intuition or his own expectations or just to please you. Can you make my sample size look better? Oh yeah, sure I can. No, that's not the way. It doesn't work like that. It's important that the clinician points out that what is clinically important for us to show, which is meaningful clinical difference. Meaningful clinical significant difference is very important to understand. So just to give an example, before you ask Khulud directly, that for instance, if you're taking, again, we talked about the fact that somebody may have dwarfism, and they may have dwarfism and they may be just at the age where epiphyses or the bones fuse, right? So maybe 12, 12 and a half or 13. And you want to give them some hormonal therapy. Now, the clinician or the endocrinologist or the pediatric endocrinologist can say, well, over a year, if this person grows by 6 inches from 4 feet to 4 feet 6, maybe that's meaningful. Maybe the person will be able to, in the future, sit in a car and reach the, accelerator, things like that. Or they might say, no, I need about 10 inches increase over this 1 year in terms of height. Now, that is where the clinician has to look at what previous experience has shown, what previous studies have shown, that if you say that, yes, you're just going to show a 1 inch difference, it may not even matter, or half a centimeter may not even matter to the person themselves. So it's important from the patient's perspective. It may not be clinically relevant, and it may, because the effect size would be so small that you choose as significant, then the sample size would really bump up to show that. But if you are looking at a higher effect size, like Khulud pointed out, 20%, 40%, 80%, something known as Cohen's D, they call it. So you can decide, is 20% meaningful and practical to achieve, or can you even make 40%? Is that achievable from previous studies? And so the sample size would come in from the clinician's perspective. What would make a meaningful difference in terms of a person's quality of life or in terms of functionality? And that is where, if you might say, okay, I want an 80% difference. I want this boy to increase by 2 feet. So unreasonable expectations will make the sample size look very small, but you're never going to achieve that. You're never going to achieve an outcome of interest. So it's extremely important that in this particular case, where she showed the 2 examples, in the recovery trial also, initially, of course, they had no idea how to go about calculating the sample size to show a difference. But then there was a recalculation based on the fact that there was nothing existing in the literature. So either you go back to the literature and see what others have done in terms of sample size calculation, or you do a pilot study. So what they did practically was they did a pilot study. And in the pilot study, they came up with the effect size or something which is meaningful in terms of the 28-day mortality. And that is where then they subsequently did the sample size calculation. All of these are sort of adaptive designs where you do sample size recalculation while the study is going on. And each of them requires going back to the IRB for protocol amendment. So questions are now open for Khulud for the next 8 minutes or so. Please, if you have any question from online that you would like Khulud to answer, please can you repeat that?
Few of the questions.
In the mic, please.
Yes, we have few questions related to how can one calculate sample size of an observational study. Khulud, can you hear us?
Can I just respond? I think
Yes.
if you can just email it to us subsequently, because we are focusing on RCTs, because that will go totally in a different direction. There are several different forms of observational studies. I would like to move on to the next question. Thank you for that. But if you email us, we can subsequently respond. Sure.
How we can take one-sided or two-sided p-value? When we take one-sided or two-sided p-value?
Go ahead, Khulud.
So a two-sided or two-tailed question means that your trial outcome can go either ways. So let's suppose you are comparing drug A with drug B. When you take a two-sided test, you are basically saying that your drug A could do better than drug B, or your drug B could do better than drug A. So it could go either side. But if you're comparing your drug with a placebo and you're very sure that no, my drug is going to do better than placebo, there's no way that placebo would do better than a drug or my drug would do worse than a placebo, then you have to take one-sided test. So there's a cost that comes with it. So whenever you're taking a two-sided test, your alpha is, let's say, set at 5% or 0.05. But when you want to go to one-sided, meaning that you are sure that either your drug A is better than B or your drug is better than intervention is better than placebo, then you have to reduce the alpha by 50%, meaning that it now comes down to 2.5% or 0.025. So this becomes more conservative and it becomes harder to prove. But in that case, you're just making a point. You're making very sure that, yes, I'm confident that my drug does better than the placebo, or drug A does better than drug B. So just to quickly recap it, you're comparing two groups. If you are very sure that one group is definitely going to do better than the other group, then you take one-sided test. But if you're unsure that either could do better than the other, then you take two-sided test. Hope this answers the question.
And just to complete the story, thank you so much, Khulud. Another thing is depending on the study design. So if you are saying that you want to do a superiority trial, then what do you think you should take in terms of the hypothesis? Two-sided or one-sided? One-sided. If you are taking, now the questions for the 500 rupees come in. Now, if you're doing an equivalence trial, two-sided or one-sided? Huh? So all those who said two-sided, 500 rupees I will take from you, but not now, in the Yawm al-Qiyamah, inshallah, later. And what about non-inferiority? One-sided or two-sided? Huh? Okay, so that's just like you said for superiority very comfortably, one-sided hypothesis. And you said for non-inferiority also, you are saying it will be not equivalent, it will be non-inferior, it will be within a certain margin. And you said one-sided, that's great. But even equivalence, you said it will be within that margin. So something just above, something just below, it is right there. This is what you are saying it will be. So you also have to go for a one-sided hypothesis in equivalence trials as well. So in general, conservatively, most people say stick to two-sided hypothesis and try and go with a p-value of 0.05. But if you are doing these kind of study designs, then by nature of the design, it will also have to be one-sided. Any other questions for Khulud for online? Or you have something here? Please. Can you pass now? Thank you. Mufassir sir.
Assalamualaikum, sir.
Welcome,
Sir, just suppose we are doing a new study, a randomized controlled on two drugs. For example, we have a painkiller, diclofenac, and a new drug that is being launched in just like 2020. So we don't have much studies in Pakistan. So there's a misconception that if you don't have studies published on that specific study you're being doing, so you need a more sample size as compared to if there are more studies published online. So you need a smaller sample size. How much true is that?
Khulud, would you like to answer that?
Yes. So that is a very vague definition. We're not using any vague definitions here. We're just using formulas. So what is the definition of more or less? That all depends on the values that you get. So let's suppose, like you said, there's a new drug that has been launched and it has not been tested before. So if you're doing an RCT, let's suppose you're starting off with phase 1. In phase 1, you obviously do not have to show the effect size. You're only studying the safety. That might be a case where there has been no previous studies done on that particular drug. But if you're starting off with phase 3, you would like the phase 2 and the phase 1 results to guide you. What did the phase 2 and the phase 1 results show you? How much was the difference or the improvement in the outcome of interest? Like how much did the pain score come down, or how many days did it take for the pain to go away? So all of these numbers, they have to go into the formula. So let's say if you're going towards the number of days, you have the mean and the standard deviation. Let's say it took 7 days plus or minus 3 with standard deviation for the pain to go away completely. or the pain score after 1 dose or 4 hours, it came down from supposing 6 to 3. Okay, so these are the numbers. If you're saying that there were no previous studies conducted, which might be in some cases that we saw in the case of COVID-19 when we were in a rush to get to a point where we had a drug with us, then the wisest thing to do is to conduct a pilot study. So in a pilot study, you would take a reasonable number between 20 to 50. You would compare your newer drug with diclofenac, and after you have collected the results from 30 or 50 subjects, then you would see that, okay, in diclofenac group, only 20% of the subjects were able to go down from moderate pain to mild pain. But in the newer drug, there were 50% people, 50% subjects where the pain went down from the moderate to the mild group. So 20% versus 50%, this would guide you. And these are the values that you would put into a sample size calculator. And the number that you come up with, that is the number that you have to use if you want your study to be adequately powered to show a clinically significant difference between your newer drug and the diclofenac.
Right,
Thank you
ma'am.
so much.
This answers it.
Thank you. We have time for one last question. You want to take it from online?
Is there any criteria which guide upon deciding which power to be used in the study?
So is there any criteria which guides which power to use in a study? I think in the interest of time, I'll just mention that initially in phase 1 and phase 2, you are okay with 60% power. Okay. And then subsequently, all phase 3 studies, there's a minimum criterion of 80%. And if you want to go up to 90%, you can go all the way up to 95 and 99%. You'll just keep on increasing your sample size, but your beta will obviously become smaller. So I think with that, Khulud, thank you so much. Again, the delightful presentation. And inshallah, I think it's hopefully at least clarify some concepts. And obviously, we don't expect you to overnight become biostatisticians, but I think it's important to understand where these sample size calculations are coming from and why it is so important to use the safe amount of patients which can answer a study with valid external generalizability and also internal validity. So it's so important to understand these concepts. Don't bank on your statistician. Don't ask the statistician to bail you out at the end of the study. You need to be able to sit with the biostatistician early on and then tell them, explain to them what is your rationale for why you're choosing this clinical significant difference. Then look up the literature earlier on and see what previous studies have shown, or conduct a pilot study, if not, and then try and figure it out. So we'll move right along, and inshallah, now you're going to be having another PharmD, Dr. Maham Zahid, again, a very respected member of the clinical research team out at National University of Medical Sciences. I've had the good fortune of working with her as well. So we are all part of a team. This is all our collaboration. So we work in different institutions now, and we just want to put things together and work together for common good, inshallah. So she's going to be taking you through inferential statistics. Again, she's not a biostatistician. So, but with the background of having been a PharmD as well as a master's in public health, certified in Harvard from Harvard in principles and practice of clinical research, and also a PhD scholar now. She has her own scholarship, and as part of that, she makes it her business to understand inferential statistics and transmit it. Thank you.
Juma bhi hai.
हमारा सबसेट 120 से ऑफ होता है 120 से 120 से पहले One quick announcement is that inshallah Maham is going to finish as promised at 1:20. Then you have 1 hour and 10 minutes. But during that time, those please, the ones who want to just have lunch out here, to be at 1:45 today. And the prayer timing is all the way between 1:20 and 2:30. Several masjids in this area for those who want to go out and come back at 2:30 for the next session. Thank you.
Assalamu alaikum everyone. My name is Maham Zahid and I'm a pharmacist and a public health professional. Currently, I'm enrolled as a PhD scholar and I'm working at National University of Medical Sciences as a clinical research associate and a lecturer of public health. So the building on the first topic, that was sample size calculation, I will be discussing inferential statistics. It is a relatively a complex topic and a lot of difficult concepts are involved, but I will try my best to simplify things for all of you, and I have incorporated only the important aspects of inferential statistics in my slides because it's not possible to cover all of the inferential statistics in 30 minutes. So let's start with the presentation. These are going to be the contents of my slides. First of all, we will see what is inferential statistics, what is importance of inferential statistics, what is hypothesis testing, confidence interval and p-value, some measures of associations that we use in RCTs, some types of analysis, commonly used statistical test, and in the end, I will discuss some case article and examples for your clarity. So basically, what is inferential statistics? As the name states, to infer. We make an inference from the sample about the population in inferential statistics. We do comparison of the outcome across the treatment groups, and we make generalizations about larger population of the subjects. So the main aim of inferential statistics is to draw some conclusions from the sample and then generalize them to the population data. This is the main difference between inferential and descriptive statistics. We do not infer from sample to population in descriptive statistics. We just describe our data. So why is inferential statistics so important? It is important because it helps us in testing the hypothesis, which is generated by observational studies. It helps us in drawing some conclusions to explain a phenomena. It also helps us in generalizing those conclusions to the population. Last but not the least, inferential statistics are helpful in predicting future outcomes. So these are four main points which are important in respect to inferential statistics. So hypothesis testing, Khulud has discussed some points about hypothesis. So I'm going to discuss it briefly. Hypothesis testing is an integral part of inferential statistics in which assumptions are made about the population parameter, and some statistical tests are then used to either accept or reject those assumptions. So as we know, there are two types of hypothesis: null hypothesis, the hypothesis of no difference, and alternate hypothesis, which is opposite of null, and it reflects the intuition of researcher, what he's going to prove. This is a normal distribution curve. So you can say that in a two-tailed hypothesis, there are two types of hypothesis, one-tailed and two-tailed. So the alpha level, it is distributed on the both sides of the tail. And in a single hypothesis, it's confined to the one side of the graph. We usually say that in clinical research, we take two-tailed hypothesis more commonly, but it's just a comment. It's not like that. So moving towards confidence interval and p-value. Confidence interval, it is basically a range of values which includes the desired true parameter or the measure of association with a probability defined in advance. We usually take our alpha as 0.05 and the corresponding confidence interval is 95%. So what does this confidence interval mean? It means that we are 95% confident that the population parameter or the measure of association that we have calculated is lying between that range. And p-value, it denotes the probability of rejecting the null hypothesis, or you can also say that it is the probability that an observed difference is due to random chance when the null hypothesis is true. So both of these, they depend upon the level of significance that we take in our study. How do we interpret confidence intervals and p-values? For example, we took significance level of 0.05 and the corresponding confidence level is 95%. Then if we calculate our p-value and it comes to be less than the significance level or 0.05, then we can say that our hypothesis test is statistically significant and there is a significant difference detected. And then we reject our null hypothesis, the hypothesis of no difference. And the confidence interval, if the range is not containing the value of null, the value of no difference, then we can say that our confidence interval is statistically significant. For example, in a relative risk of 3.5, confidence interval is 2 to 5, and a p-value is less than 0.001. So what does this mean? A relative risk of 3.5, it means that people who are belonging to the intervention group or who are exposed to an intervention are the rate of occurrence of event in those people is 3 times more likely to occur as compared to control group. And this 3.5 value, it can lie between 2 to 5, it can vary. And the p-value is also significant as it is less than 0.05. So it's a significant difference exists. Okay, now let's discuss about some measures of association. Basically, measures of association are used to quantify the strength and direction of relationship that exists between an exposure or an intervention and outcome variables. And it enables comparison between different groups. So there are many measures of associations. In randomized control trials, the most commonly used measures of associations, they include a relative risk, which is the ratio of incidence in the intervention group to the incidence in the control group. And we denoted it by RR, and RR of 1, the relative risk of 1, it means that the risk of outcome is equal in both the groups. There is no difference in intervention group and the control group in terms of risk. And when the relative risk is less than 1, we can say that the intervention has a protective effect on the patient. The patients who are belonging to the intervention group are less likely to experience the event of interest, and similarly vice versa. So the second measure of association is absolute risk reduction. The absolute risk, it shows the absolute difference in outcomes between one group, usually the control, and the group who is receiving the treatment. We can calculate it as control event rate minus experimental event rate. And the equation shows that ARR is equal to CER minus EER. Another measure of association is relative risk reduction, and it shows how much the treatment reduced the risk of bad outcome that would occur relative to the control group. And it is calculated as control event rate minus experimental event rate, which was absolute risk reduction, divided by the control event rate. So it is an extension of absolute risk reduction. And this is its formula. So the next measure of association is hazard ratio. We use hazard ratio when our outcome of interest is in terms of time to event. And it is the ratio of rate at which patients with intervention experience an event to the rate at which patients without intervention experience an event. It is similar to relative risk. We also interpret it like we do relative risk. For example, like hazard ratio of 1 means there is no difference between the occurrence of hazard in both the groups. And if it's less than 1, then we can say people who are belonging to the intervention group are less likely to have hazard, or the risk is less than the control group. Another measure of association, which is very important, is number needed to treat. It shows us the number of patients who must be treated for one patient to benefit. For example, if our number needed to treat is 20, it means that we need to treat 20 people in order to get benefit for one patient. The one patient will definitely get benefit from the treatment. So it is calculated as 1 divided by the absolute risk reduction. So the confidence intervals, they are calculated for each measure of association in order to quantify their uncertainty. For example, as discussed previously, relative risk of 0.8 with 95% confidence interval of 0.5 to 2.5 and a P of 0.1. So the relative risk is less than 1, so it's showing that people who were belonging to the intervention group or who were exposed, less number of events were occurring in such people. But please note that confidence interval, it's ranging from 0.5 to 2.5. The value of 1 is coming in between. So this confidence interval, it is not significant. And similarly, the p-value, it's 0.1. So it's more than the alpha level. If alpha level is 0.05, so this result is not statistically significant. On the other hand, if we see hazard ratio of 1.5 and a confidence interval of 1.5 to 1.8, then the value of hazard can lie between 1.2 to 1.8, and p-value is significant. So confidence interval is also significant and p-value is also significant. This is just for example, it's hypothetical. Okay, now there are 3 types of analysis in RCTs. I'm going to discuss all three of them. I'm going to just define them. So a per protocol analysis. In a per protocol analysis, comparison of treatment groups includes only those patients who completed the treatment originally allocated during randomization. If done alone, this analysis leads to bias. Why? Because it removes the data from patients who did not comply with the protocol. So what we do, we remove those patients who failed to comply with our intervention. If a person was randomized to intervention, but he didn't take the medicine or he didn't comply with the protocol, we will remove his data from the study when we will analyze the results. So that's why we say it's not a robust design. It can lead to some bias. Similarly, there is another type of analysis known as treated analysis, in which the participants, they are analyzed according to the treatment that they received rather than according to the treatment that they were assigned to. For example, a patient was assigned to intervention, but he failed to comply. So at the end, when we will analyze the data, we will consider him in control group rather than considering him in the intervention group. So we are not removing the data, but we are shifting the patient from one group to another while analyzing the data. So this design is also biased. It can lead to false results. Then we have intention to treat analysis, which is a robust analysis method in which the patients are analyzed according to the groups to which they were originally assigned. You can also say that once randomized, always analyzed. If a patient was non-compliant, irrespective of his compliance, he will be analyzed in the same group. So this is the most efficient method of analysis. Okay, so what is a statistical test? Why do we perform a statistical test? Basically, the purpose of conducting a clinical trial is to compare efficacy of a new preparation or a drug with efficacy of an established preparation or a placebo in a control group. And we want to know whether the observed difference between the two treatment groups are just at random or are they really present. So in order to do that, we perform statistical tests to prove those differences. And there are some steps that are to be followed in performing a statistical test. These steps are stating the question to be answered or the research question that you want to answer, its statement, formulation of your null and alternate hypothesis. Step 3 is decision for a suitable test. You need to choose from a pool of tests that which test are you going to perform in your study. And then you specify the level of significance, if it's 0.05, 0.01, whatever it is. And then you perform statistical test analysis, calculate the p-value, and you make a statistical decision based on the p-value. If p is less than 0.05, then you say you reject the null hypothesis and accept the alternate one. And if it's more than or equal to 0.05, then it leads to retention of the null hypothesis. We fail to reject the null hypothesis. And in the end, we do interpretation of the test result. So these are the common steps that are used in every type of statistical test. So some types of statistical tests, there are various types of statistical tests that we use in research, and the decision to choose a specific test, it depends upon 3 important parameters. First of all, the research question that you want to answer. Secondly, the data structure, the type of variables that you are including in your study, dependent variables, independent variables, either they are continuous in nature or categorical. And thirdly, the study design. The study design is also important. All these 3 parameters are important before you decide what statistical test is to be done. There are different types of tests, and they can be generally categorized on the basis either they are parametric or non-parametric tests. So a parametric test, it makes assumption that the population data is normally distributed. We apply parametric tests when we are sure that our data is normally distributed. And on the other hand, non-parametric tests are distribution-free tests, and they are used when our data is not normally distributed or the sample size is very less, like for example, less than 30. On a less than 30 sample, you use non-parametric tests. So let's discuss some commonly used statistical tests. On the basis of statistics that is to be performed, we can divide the statistical tests as comparison tests, correlation tests, regression tests, and survival analysis. So these are 4 categories of tests based on the statistics that is to be performed. For example, in comparison tests, we compare the mean outcome mean between the two groups, intervention group and control group. And the outcome variable is usually continuous in nature so that we can use its mean. And the independent variable, either it's grouped like intervention and control group, and we compare the mean. The non-parametric equivalent of pair t-test is Wilcoxon rank, and we used like pair t-test means that the data is taken from the same individual before and after the intervention. Then we have independent t-test. The samples are independent of each other. Non-parametric equivalent of which is man Whitney U. And similarly, the mean is compared between the two groups. Then we have one-way ANOVA or MANOVA, MANCOVA, which can be used when the categories are more than two, when we have more than two groups and we want to compare mean between them. And the non-parametric equivalent of which is called Wallace test. So these are comparison tests. Then we have correlation test when we want to correlate two variables. Like for example, if we have two continuous variables, one is age of a child and other is weight of a child. Both of the variables are continuous in nature and we want to see how they are correlated with each other. So then we will use a Pearson correlation or a Spearman correlation, depending on the nature of the data. And secondly, if we have both the dependent and independent variables are categorical in nature, then we use chi-square test or Fisher exact test. Thirdly, we have regression tests. There are many types of regression tests like simple linear regression, multiple linear regression, logistic regression, and multinomial. It depends upon the nature of our outcome variable, which type of regression is to be adopted. For example, in simple linear, the dependent and independent variables are both continuous in nature. In logistic regression, we have the outcome variable in terms of a binary outcome, yes or no, death or alive. So there are different types of regression tests. Fourthly, we have survival analysis. And in survival analysis, we have Kaplan-Meier curves, which gives us a visual depiction of the survival time and survival rate between two groups. And there is a log rank test. It's not mentioned here, but we use log rank tests to compare the survival rate between the two groups, if it's significantly different or not. And then we have Cox regression. It's a regression method used in survival analysis when the outcome event is in terms of time to event. This is important thing. So just to summarize, what is survival analysis? Survival analysis compares two or more groups with respect to time. Time is the most important thing in survival analysis when the primary target parameter is time until an event occurs. So our outcome variable is in terms of time. For example, time to heart attack, time to cancer remission, or time to death. So what type of research questions are answered by a survival analysis? I have included 3 examples here. The research question can be, what is the probability that a participant survives 5 years? Are there differences in survival between groups, between those assigned to a new versus a standard drug in a clinical trial? How do certain personal, behavioral, or clinical characteristics affect participants' chances of survival? So these are the types of questions that we usually answer with the help of survival analysis. And survival analysis focuses on 2 important pieces of information. Number 1, whether or not a participant suffers the event of interest during the study period. If event occurs, we code it as 1, and if event does not occur, we code it as 0. And secondly, the follow-up time for each individual being followed, for how long an individual was followed, who was enrolled in the study. So these are the 2 important pieces of information that we note in a survival analysis. Then we have survival curves, also known as Kaplan-Meier curves. These curves are plotted as step functions. The time is on the x-axis, and the survival is given on the left side, the y-axis, and also known as Kaplan-Meier curves. And these curves are used to compare the survival times and survival rate between groups. So what is the survival rate? Survival rate basically indicates the number of people in whom no event has occurred up to a certain point in time. And the median survival time is the time at which half the patients have suffered an event. So this is an example of Kaplan-Meier curve. You can see that on x-axis, on the bottom line, we have time from primary surgery to death in years. And on the left side, on the y-axis, we have survival time rate. At 0 time, both the groups, the patient belonging to both the groups had maximum survival rate. Okay, so there are two groups. The brown line shows maintenance chemotherapy group and the blue one shows sandwich chemotherapy group. So at time 0, both the groups, they started at maximum survival. And as the time went on, the graphs are coming down in a stepwise fashion. As soon as the patient died, the graph, it came a step down. So it is clear that the blue line, the blue graph is coming downwards really quickly as compared to the brown one. So the survival rate is better in maintenance chemotherapy group as compared to the sandwich chemotherapy group. Then there is a log rank test. It examines whether there is a significant difference between two group survival time or not. And it gives us results in terms of hazard and hazard ratio. So hazard is the death rate, and hazard ratio states how much higher the death rate is in one group as compared to the other group. The hazard ratio, it is a descriptive measure, a measure of association that is used to compare survival times of two different groups of patients, and it is interpreted same as we interpret relative risk. And then we have Cox regression. It is a type of regression when the outcome variable is in terms of time, and it is a multivariable analysis. So simultaneous effects of several variables on the survival time can be investigated with the help of Cox regression. Okay, now let's discuss some of the case articles. The first case study is, the article is dapagliflozin in patients with chronic kidney disease. Our research question in this research was, is dapagliflozin effective in patients with chronic kidney disease to reduce the decline in GFR, risk of end-stage kidney disease, or death from renal or cardiovascular cause? And the study design was randomized controlled trial, double-blinded. The primary outcome, the outcome variable was in terms of time to event, and it was a composite of three things: decline in GFR, end-stage renal disease, and death. So it stated in their statistical analysis section under methodology that intention to treat analysis was performed. So it's a plus point. And then they used Cox proportional hazard regression method because the outcome event was in terms of time. So they have used survival analysis in their analysis. So, and it was stratified according to the factors used at randomization, that was type 2 diabetes was present or not. And then the measure of association that they used is hazard ratio and a 95% confidence interval. They also summarized the safety data, and the software that they used was SAS software. So in the results, they they give that the primary composite outcome, it was found that patients who were belonging to the dapagliflozin, the intervention group, 9.2% of those patients, they suffered the outcome of interest, as compared to 14.5% who were belonging to the placebo group. So clearly we can see the percentage of placebo group is higher than the intervention group. So more number of outcomes are occurring in the placebo group. And in terms of this measure of association, the hazard ratio is 0.061. It is less than 1. So we can say that the intervention has a protective effect in terms of the outcome in patients. Less number of patients who were in the intervention groups are suffering or experiencing the outcome. The confidence interval is 0.51 to 0.72. It is also significant because it's not containing the value of 1, and the p-value is also significant as it is less than 0.001. And another measure of association that they have given here is number needed to treat. So the number needed to treat was 19. So if we treat 19 people with intervention, there is a chance that we will benefit at least one patient. So it's a good number. Then they have said that the results were consistent throughout the study groups. Like the patients who were diabetic, hazard ratio was calculated to be 0.64 in patients who were diabetic. And patients who are not diabetic, the hazard ratio has been further reduced, which is even better, 0.5. So it means that the chances of outcome were 50% more likely to occur in the control group. And confidence interval is 0.35 to 0.72, which is significant. P-value is not given here, but it is clear that these results are significant. So these are the graphs taken from the article, Kaplan-Meier curves. The red line is the placebo line and the blue one is dapagliflozin line. We can see that more number of events are occurring in the placebo group. You can see that on the y-axis, we have cumulative incidence. So more number of events are occurring over time in the placebo group as compared to the dapagliflozin group in both the graph A and B. Similarly, in graph C and D, when composite of death from cardiovascular cause or hospitalization and death from any cause. Similarly, the placebo graph is going more upwards as compared to the dapagliflozin. So more number of deaths are occurring in the placebo group. The hazard ratios are also mentioned on the graph, 0.71 and 0.69. So both of them are less than 1. So the intervention has a protective effect. Okay, this table gives hazard ratio according to the various subgroups. You can see that all of the hazard ratios, they are on the left side from the dotted one. So when they are less than 1, intervention is clearly benefiting all of the patients in all the subgroup analysis. And in the end, it is concluded that the intervention was beneficial and less number of events occurred in people who were belonging to the intervention group as compared to the placebo. So our other article is the recovery trial, dexamethasone in hospitalized patients with COVID-19. So the research question was, is dexamethasone effective in reducing 28-day mortality among patients infected with SARS-CoV-2 compared with treatment as usual? And study design was randomized controlled trial. Outcome variable was again in terms of time to event. It was 28-day mortality, the primary outcome. So in the method section, they have mentioned under statistical analysis that for primary outcome of 28-day mortality, Cox regression method was used to estimate the mortality rate ratio, and Kaplan-Meier curves were used to show the results. And the measure of association was risk ratio, or you can say hazard ratio. And it was compared between the outcome of recipients of non-invasive and invasive mechanical ventilation. The patients who were on invasive or non-invasive mechanical ventilation or recipients of renal replacement therapy. So a comparison was made. And the risk ratios, they were adjusted for baseline age. So there was a difference between the mean age of intervention and control group. So that's why they have adjusted their ratios in order to remove the age bias from the results. And then intention to treat principle was followed. Okay, so in results, they say that mortality at 28 days was significantly lower in the dexamethasone group as compared to the control group. The percentages are 22.9 versus 25.7, and the rate ratio, the measure of association, it states 0.83. So it's less than 1. So dexamethasone has a protective effect against occurrence of primary outcome. Confidence interval is also significant, and p-value is also significant. So they say dexamethasone, in the dexamethasone group, the incidence of death was lower than in the usual care group among patients receiving invasive mechanical ventilation. So people who were on mechanical ventilation and receiving the intervention, less number of people were suffering or experiencing the event of interest or the outcome, 29.3% versus 41.4% in the placebo group or the control group. So the rate ratio is 0.64, less than 1, and the confidence interval is 0.51 to 81. And the people who were not receiving oxygen without invasive mechanical ventilation, people belonging to intervention group, 23.3% were experienced the primary outcome versus 26.2 in the control group. And the rate ratio was 0.82 with the confidence interval of 0.72 to 0.94. And people, this is an interesting finding. People who were not receiving any respiratory support, who were not on oxygen therapy, more number of events were occurring in patients receiving dexamethasone, 17.8% versus 14% in the control group. So dexamethasone was not helping such kind of patients at all. And the rate ratio is 1.19, which is more than 1. So it means more number of the primary composite outcomes was occurring in the intervention group. And the confidence interval is 0.92 to 1.5. It's containing the value of 1. So it's not, the finding is not statistically significant, but it is an interesting finding overall. So this is the table taken from the article. There are two groups, dexamethasone and control group, and the rate ratios are compared according to the primary and the secondary outcomes. And this is the Kaplan-Meier curve for all participants and for invasive mechanical ventilation. You can see that in all participants, when the number of people, the sample is 6,425. The dexamethasone, the red line, is a little bit lower than the control or the usual care group. So less number of events are occurring in the dexamethasone group. But the difference is not so much. It is the statistical significant difference, but a little minor difference is there. Whereas in invasive mechanical ventilation, you can see the graphs are more apart from each other, the usual care and the dexamethasone graph. So the dexamethasone, it is giving more benefit to people who are on invasive mechanical ventilation as compared to the control group. And similarly, who were only on oxygen and people who were not on oxygen, who were not receiving oxygen. You can see in the fourth graph, the dexamethasone graph, the red line is crossing over the control group. So more number of events are occurring in dexamethasone group. So in the end, it is concluded that in patients hospitalized with COVID-19, the use of dexamethasone resulted in a lower 28-day mortality among those who were receiving either invasive or mechanical ventilation or oxygen alone at randomization, but not among those receiving no respiratory support. So dexamethasone is not beneficial for mild to moderate type of COVID disease. So that's all from my side. And you can ask questions or post your queries to the mentioned email IDs.
Thank you so much, Maham. Again, I understand that not all of you are biostatisticians out here, but you will appreciate the fact that once you start understanding literature, once you start getting into the study designs, then sample size calculation, and then the inferential statistics that follow, they also follow a certain pattern depending on how the data looks like. And each statistical test is chosen with respect to what is the study question, the study design, the alpha, beta, and whether it is two-sided, one-sided, and then whether the data is symmetrically distributed or asymmetrically distributed. Whether you have a pre and post comparison, or whether you have two groups being compared, or whether you have more than two groups being compared, all of them have different statistical tests. So as long as one is familiar that this is how this is done, you don't specifically have to do this portion yourself. This is where your biostatistician comes in and helps you out in determining what would be an appropriate test to put together. And then obviously you also can then, if you understand this, you will also not get misled. But once you are in the field of clinical research, all these articles that are being published, 90% of the articles that are being published in the New England Journal of Medicine have survival analysis. And if you have a visual idea of what it means, where you show curves going up and down, it's just a cumulative probability which you did in maybe high school. So if somebody survives to a certain point and the chance of survival is 90%, and then to another point, the chance of survival is 80%, so the cumulative survival is 90% into 80%, it's 72%. If somebody then has a survival of another 50%, the cumulative survival is 36%. It's just mathematics. Just go up and down, and where events are not observed, that's what's known as censoring in the context of survival analysis. And I think, again, a detailed statistical 3-day workshop might give you a little bit more clues on something like that, where you can do things hands-on. So our idea was to put things together. Maham, you have any online questions for Maham? Yeah, Dr. Rehma? We have exactly 2.5 minutes before we close, and then inshallah, those who want to have lunch, it will start off at 1:45. Those who want to go for prayer, there are several masajid in this area. The prayer is usually around 1:50, 2 o'clock, and then we will reconvene at 2:30, inshallah. So you have any question quickly that you can?
Does survival analysis focuses on natural history and for chronic diseases?
So the question is, does survival analysis focus on natural? So yes, you can use survival analysis even in cohort, in prognostication as well. So in cohort studies also, you can use survival analysis, just a time to event. So time to event or survival simply means that, it's a cumulative probability of achieving an event or surviving from a certain point till the observation time is over. And so if you're looking at natural history, in obviously in observational studies, you do not have any intervention. You're just looking at an observation of a cohort. And then you can have controls there as well, but you can just see the natural history and plot it against time as well. So if you're looking at survival of patients who voluntarily smoke and you're not doing any intervention, and those who don't smoke and you're doing survival curves, you can do that. So obviously, you're not going to do a randomized control trial for that. But yes, in natural history, survival curves can also be easily used. Any other question?
This would answer our next question as well. In what epidemiological study survival analysis can be done? So it can be done.
Please see question number 1.
Cohort.
Next one.
One of our participants has said, I'm lost in what is a composite outcome.
Okay, so that's my fault from yesterday. So just to give you something quick and straight, that in general, you want to look at one primary outcome measure, but you will a lot of times have several secondary outcome measures for any particular study. The one primary outcome measure itself, if it's a single outcome measure, like mortality or myocardial infarction or end-stage renal disease occurring, that's all single primary outcomes. But if I want to say that I'm not sure if I can show a mortality, but what if I say if somebody has mortality or a myocardial infarction or end-stage kidney disease or stroke, all of them are important clinically, and if I put them together in a composite outcome, then the first one of any of those 4 occurring will be the endpoint. That's all a composite outcome means. The only problem is you have to have your sample size accounted for all those 4 outcomes when you're putting it in a composite outcome. So, but the first one, any one of that occurring is what is the composite outcome. And several times you have lots of people putting in and jumbling together lots of composite outcomes. Some of them may not be clinically important. And that is where you can get misled, or the researcher may be trying to mislead. I don't want to say that, but they're trying to show some effect. And even if it's not clinically important, that's where you as a clinical researcher or those who look at literature critically are supposed to look at it, all outcomes clinically important. And when we do this EBM session this afternoon, Professor Mubin Iqbal will do that. He will mention what are clinically important outcomes of interest. And I think this is where this will be revisited. Any other question? Last question online? No, we're out of time. Bam. So that's it. Thank you very much. And inshallah, we'll see you at 2:30 and enjoy your Jummah prayer, enjoy your lunch. Thank you. Assalamu alaikum. Thank you, Regime. Thank you very much for coming back. So we are going to go through 11 slides. 2 of them have just labels and 9 of them have the consort statement. So please don't go to sleep through it, because I'm going to try and inshallah take you through a little bit of what we've done from yesterday till this afternoon. And hopefully things will fall into place, that when you want to conduct a randomized control trial, there are several different ways one can conduct it with modifications. Obviously, we have mainly focused on a parallel group superiority kind of trial format. And a lot of times people were finding the inconsistency in reporting to be a major hurdle in reproducing studies, because a lot of times you want phase 3 studies to be reproduced at least once before a new drug is approved. Mostly you need 2 well-conducted superiority randomized control trials over placebo to introduce a new drug or approve a new drug. And in this context, when you don't have standardization, sometimes the readers get confused. They don't know how to interpret results. So we have also tried to keep to a certain format, looking at the consort statement, and that's how it's expected that one reports the results of the randomized controlled trial. So this was first developed, the consolidated statement for reporting trials or randomized trials. It was first developed in 1996 by Schultz et al. And the group is based in Canada. And then they revised it in 2001 and 2010. And if you go on the website, you'll find Schultz everywhere, where they have come up with modifications to include pragmatic trials and to include other extensions of the standardized randomized controlled 2 parallel arm group trial. So what are the elements of this trial? And out of the 3 studies that we have provided to you, which one comes closest to following this particular format? Not saying this is the only way one has to do it, but a lot of journals probably expect it now, and the diversity has been lost, but at the cost, at what cost? Perhaps they've gained some standardization through that. So when we looked at the DAPA-CKD and the recovery trial, as well as the HIP attack trial, it seemed like the HIP attack is closest to following the consort statement. And so we are going to use examples from that. And the other 2 were more of an adaptive design recovery trial, where they had to change design midway with all this pandemic and pressure of mortalities with COVID. And the DAPA-CKD, the US group and the Canadian groups, usually they have their own ways of reporting things. Sometimes one side does not want to follow a certain conformist thing, which is okay. Then there's the Lancet group, which has a little bit more diversity in the way you report things. But for most practical purposes, for learners and for those who want to get into this business, the consort statement would be useful. So one of the things, so they will go through the entire list. So if you look at this, starting from title and abstract to introduction to methods with trial design, participants, interventions, outcomes, sample size, randomization sequence, allocation concealment, implementation, blinding, participant flow, baseline numbers analyzed, outcome estimation, statistical methods, all those things are put together in a protocol, which if you follow it at the beginning, when you're designing the trial or you're part of a team, you obviously will not do fishing expeditions. Subsequently, you will be able to plan the trial at the beginning and report it accordingly. So if you put together a good proposal, which and then details the protocol, 2/3 of a manuscript is practically done. You just have to add the results, a few things in the methodology, and then talk about interpretation in your discussion. So let's just go through this checklist. So they ask for the title and abstract to have identification as a randomized trial in the title. So the HIP attack trial actually does state that this is a randomized controlled trial of rapid versus usual care in terms of addressing HIP attack or hip fractures presenting to the emergency rooms. They also say that in your abstract, obviously, that is a pretty standard abstract. You talk about a trial design, a little bit of the gap statement, the methods and the results and the conclusions, and that is pretty much standard. That I don't think the consort is stating anything different from the usual. And in the introduction, they want you to talk about the scientific background and explanation of rationale and the specific objectives or hypothesis. So if you just simply look at what I've written here, I've said that yes, the HIP attack trial was identified as an RCT within the title. And then comes the important things in the introduction and background. So normally when you're putting an introduction and background, if you have 3 paragraphs, that's enough. One is what is known. And second one, starting with the word however, what is not known. And the third paragraph starting with therefore, the rationale for doing a study. If you do this, that's all you need in an introduction. It can be 1 page of typed double-spaced page of a New England Journal of Medicine article or any other article. That's all one needs to do. In introduction, our background obviously has to be related to the question of interest and not the whole background of the entire world, with different kinds of hip fractures and this and that. No, you just want to say that yes, and hip fractures presenting with, to emergency rooms with low intensity hip fractures, low energy hip fractures, uncomplicated hip fractures, then, previous observational studies and some other designs have demonstrated that if you take them to the operating room earlier, you may have lesser complications with thromboembolic and cardiovascular complications. A lot of patients with hip fracture, because of comorbidities, unfortunately, they're likely to develop. So you talk about that particular background. And so they're saying, just stick to the scientific background and explanation of the rationale. So you're building up a rationale in the introduction. This is known, however, this is not known. Therefore, we, with this particular hypothesis or this particular background, are planning to do this study. So that would be at the beginning. The title and abstract, usually, whether you are performing a study or reporting it, usually should be done at the end. So titles obviously would have to be very, very carefully chosen subsequently at the end to actually spoke. But they say in consort, if you're doing an RCT, identify it within the title that this is an RCT, a randomized control trial of this versus that, a randomized control. Does aspirin compared to placebo decrease the risk of stroke in patients, with such and such background? A randomized control trial, or a randomized control trial of whatever interventions versus the comparator. So that is what would come in that. And then the specific objectives or hypothesis can always be built in. You don't always have to state what your null hypothesis or alternate hypothesis is when you're reporting results. But a lot of people would try and fit that in as well, that, the our null hypothesis was this and that. But that usually for beginners is recommended. And a lot of people assume that with the protocol and proposal that they have put in, the hypothesis is pretty obvious based on whether it's a one-sided or two-sided hypothesis. Then the other thing that comes in is a trial design. So in trial design, besides saying that it's a randomized control trial, one has to clearly state it's a randomized control parallel group 1:1 trial. It's a randomized control trial parallel group 1:1:1 trial. So you can have multiple groups in comparison. Or you can say it's a randomized control parallel group crossover trial. So a crossover trial for where subjects serve as their own control has to be then identified. Or you talk about what is known as a factorial design. And in a factorial design, we did not go through that to make things any more complicated, but it's just that multiple levels of different factors are then adjusted within the trial design so that you can study two or more different either doses or two or more different combination of drugs with some usual care elements at the same time. The only problem with factorial designs is that because although you can reduce the sample size to study a few things extra, but you have what is known as interaction effects between things which are being allowed simultaneously as factors in the study. Then when we are talking about eligibility, so eligibility criteria for participants has to be clearly, okay. Then they say important changes to methods after trial commencement, such as eligibility criteria with reasons. So if during the trial you had to recalculate your sample size because there was some element of adherence or more dropouts than what were anticipated, so then those things need to be clearly stated when reporting. Because after all, this is about reporting. This is not about generating a proposal or protocol, about reporting trials. So you talk about the eligibility criteria, and if they were changed, why were they changed? Just like in this recovery trial, they did not use pregnant women initially, thinking of some confounding or whatever effects they must have thought, and then they started seeing pregnant women dying of COVID, and then they included them. And the denial of dexamethasone was obviously not something that they probably could justify subsequently. Participants' eligibility criteria, we talked about it yesterday. The same inclusion criteria, whom to include, why to include, whom to exclude, why to exclude. So eligibility criteria, the inclusion criteria will be there, whether it is age related or gender related or the background or the condition that we have. And where is the trial going to be done? The settings and locations. So it's in a multi-center trial, outpatient, for 40 centers, 30 centers, 30 sites. So that like this hip attack, I believe, I think there were about 46 or 47 sites. And one of the sites that we were part of when I was at Shifa was, I think, identified as what, site number 42 or 46, something like that. So you have to identify that which sites you did that trial at. Now, when we're talking about interventions, so the interventions obviously have to be described in sufficient detail to allow replication. So after all, what is research? To see what everybody else has seen and to ask what nobody else has asked before, right? So Borji said that once. So one needs to look at things and be able to replicate findings. It's always important that you have an open mind. That, remember, we said that a research question is simply answering whether you accepted the null hypothesis or rejected it. There's no final answer because you never sampled the entire population, which you can't. So it's important that replication is done. So two independently verifiable randomized controlled trials, superiority showing superiority to placebo, is what agencies like FDA and others require to approve a new drug, because not that they don't trust it, but they just want the findings to be replicable by others. So that is why the details have to be mentioned in sufficient clarity, with sufficient clarity for others to be able to replicate your findings. Then they talk about the outcomes. Okay, now in the interventions as well, when were they actually administered? So the specific times at which interventions were administered, or specific times some assessment of outcomes were done, all those things have to be linked together with the time frame of your trial, the way it progressed. So these medications were administered, or this intervention was done at weeks 3, 5, 7, 9, 11, whatever has to be specifically mentioned. In terms of outcomes, so they want you to report completely defined pre-specified primary and secondary outcome measures, including how and when they were assessed. So when we talk about the word a priori, it means that the researcher should not go with a fishing expedition. They need to define the outcome measures before they set out to do a study, and those outcome measures should be clearly defined as primary and secondary, as the case may be. You can have one primary, you can have two primaries, you can have a composite as a primary, you can have surrogate outcomes as the primary outcome measure. But when you're talking about both of them, primary and secondary, they should be pre-specified. And how and when they were assessed, at what times the outcomes were assessed. At 3 months into the at completion of data, at 3 months follow-up, at 6 months follow-up, all those things should be completely described. Any changes to the trial outcomes after the trial commenced with reasons. So one of the things with this, I believe, Remdesivir's trial, Dr. Mubin, what was it called? The one which FDA did, Remdesivir one, the one which Fauci and team came out. So I don't remember the name of the trial, but in the Remdesivir group, initially they said we are going to show an improvement in mortality, a hard clinical endpoint. And then they somehow, something happened, I don't know, through intuition or I don't know what was the endpoint. They decided that we're just going to look at time to hospitalization or time to discharge. And initially they were trying to sell Remdesivir as the be all and end all of everything, but then they ended up coming out and saying, oh, this trial showed that we decreased hospitalization by 4 days, from 13 days to 9 days or something like that, if I remember correctly. So they changed the outcome measure during the study, and that kind of leaves you with a little bit of a you feel that I mean, like some kind of they're trying to show some results of significance, and that's where the statistics sometimes gets its bad name for lies and damn lies. It's statistics. It's not entirely that, we know that. Okay, then how sample size was determined. So exactly as Khulud showed you today, how sample size was determined. You have to describe that we used a power of 90% using an alpha of 0.05, a beta of 10%, using a two-sided p-value of 0.05 to estimate an improvement or improvement in hazard ratio or from, or reduction in hazard ratio or improvement in risk ratio of 30% in two groups to estimate this sample size, accounting for 20% dropouts. So this is how it has to be reported as a complete statement in your sample size description. When applicable, explanation of any interim analysis and stopping guidelines. So we had mentioned in our quiz yesterday that people like to do interim analysis, not because they want to, but a group of bioethicists, clinicians, mostly biostatisticians get together at the back of any intervention trial where there's more than minimal risk. So whenever there's more than minimal risk, where your IRB has to have a full body looking at a trial, and you are concerned about safety or an early efficacy signal, and you think that the study results, if you wait for a year or year and a half or 2 years, might be too late down in the line. So without breaking the blind of the investigator and without breaking the blind of the outcome assessor or the patient, there is an independent data safety monitoring board behind the screen, which is following your trial. And they pre-specify that they will look at outcomes and do interim analysis. The investigators have to specify at certain points during the trial. So they might say, we will do during these 1.5 years, 3 interim analysis. Or during these 1.5 years, we are going to do 4 interim analysis. So each time you do an interim analysis, because you are not powered to answer a question, you might sometimes miss some important findings. And sometimes because clinical trials move a little bit to the right, a little bit to the left, and then they become stable on one side, either benefit or harm. So initially when they start off, the stability of the trial is dependent on the numbers. When the numbers start getting bigger and bigger and bigger, more power, then the trials home in on one side of the point of no effect. So sometimes when you stop too early, you might make a false positive thing. So you might find things are working, and then if the trial has continued longer, they may not work. So there is a price to pay. So that alpha of 0.05 has to be then cut down, and your p-value for significance will come down from 0.05 to some other level. One of the most common ones they use is called Habital-Peto method, where they simply the price you pay is 0.01 for each interim analysis. So if you do 3, your p-value will be not 0.05, it will become 0.047. That's one of the simplest ones and the most commonly used, but there's other one called O'Brien-Fleming that is also sometimes used. But one needs to know that. That's a superstitious job, but one needs to know that. When applicable, so when did you do that, and what are the stopping guidelines? So stopping guidelines, if you have a pre-specified efficacy outcome that for benefit, you will stop at this level, or for harm, when this level is reached, you will stop the trial. Then the trial has to be stopped because certain harm or benefit has been achieved, and it would be unethical to continue to do the trial on other subjects when you already have a pretty decent benefit early on or a significant harm earlier on. So in this particular thing, I comment on HIPAA. I mean, these are not expert comments. This is just my own interpretation that all aspects in methodology section follow the above. It's a parallel group RCT with clearly defined eligibility criteria. The emergency room settings in multiple sites internationally identified. It states exact interventions and co-interventions in both accelerated and usual care limbs. This is the HIPAA trial. It mentions 2 primary outcomes, all-cause 90-day mortality and a composite of perioperative complications, various perioperative complications. Any one of them, if it occurs, then it's an outcome has been achieved. Sample size was calculated based on a hazard ratio of 0.7 and adjustments based on multiple comparisons with adjusted type 1 errors for the alpha, which is also usually known as something known as Bonferroni correction, but there are other forms for interim analysis, but this was done in this trial. Then in the randomization sequence generation, they want you to say method used to generate the randomized allocation sequence. Was it a random numbers thing, the older times? No. Was it a web-based randomization interactive system? Usually telephone-based randomization, a lot of times. So these are the kind of things that you have to state. How was it generated? And then the type of randomization. So you talk about any restriction, whether whether it was in block sizes of 3, 6, 9, or whether it was in permuted blocks, or whether it was stratified according to characteristics. All those things have to be specified in the randomization that Dr. Rahma explained to you yesterday. Mechanisms used to implement the random allocation sequence. So there's sequentially numbered containers. So what Dr. Imaduddin yesterday was showing to you, those two envelopes, there's an actual method in that allocation concealment which talks about SNOWs. So sequentially numbered, opaque, sealed envelopes, SNOWs. And that is, if that's what you use the sequence within those envelopes that you cannot hold up to light and break the code, then you need to identify that that's what you use, or some other labels in various containers that this medication or that medication. But in this particular case, allocation was concealed. Who's going to go to the accelerated limb? Who's going to go to the normal limb of usual care? So accelerated limb meant that those who were in the emergency room, within 6 hours, they had to be shipped off to the operating room with all the medical and cardiology and other clearances and reversing the anticoagulation if they were on. So lots of things had to be done within 6 hours. And the other group could do that at the usual care, which could be about over 48 hours. So all those things needed to be, including the allocation concealment needs to be described till the interventions were assigned. Then who generated the random sequence? So the statistician or central web registry or an automated voice response system generated the random sequence is what needs to be mentioned. Who enrolled the participants? So, who are the ones who investigator number 4, 5, 6, or the clinical research associates who enrolled the participants? Who assigned participants to interventions? So, of course, once you have a certain sequence held in central pharmacy or whatever, then somebody has to open those envelopes and decide what the sequence is. Or you call the central web registry and they will tell you that this patient needs to go on that. This is how the allocation is. So then who assigned the participants? Who was the person responsible should be reported or at least have a record of that. Blinding, if done, who was blinded after assignment to interventions, participants, caregivers, and those who are assessing outcomes and how. So like we said, you can have multiple levels of blinding. So what levels were achieved? And if relevant, description of the similarity of interventions. So usually you want to make sure that the readers or the clinicians out there who are going to apply that evidence to practice need to be convinced that that it's just because of that intervention which is different in limb A and B that your outcome is showing up as a different outcome. And so all the core interventions were balanced. So if this group was on ACE inhibitors, this group was on ACE inhibitors, almost equal. This group was an anticoagulant, this group was an anticoagulant, almost equal. This group had people over the age of 70. This group also had similar numbers. So by good randomization, you should be able to distribute fairly evenly the baseline risk factors. But some people are on certain things where the randomization shows you an imbalance. So even just by virtually looking at it, you can see that the groups are not balanced, or you have a p-value, which some people still report. You don't have to show that, yes, that the groups were at this baseline characteristics were not evenly matched, or randomization did not evenly distribute them. So those things need to be mentioned so that we are convinced that, yes, it's just this intervention versus that intervention which led to a difference in outcome. So here I just comment that HipAttack used a web-based randomization with varying block sizes and stratification. The varying block sizes, Dr. Rahma very nicely explained yesterday, to prevent you from guessing the next in the sequence. If they are fixed, then you have problems. If they have varying, you can't guess. And stratification if you need to stratify for diabetes or other things in there. So that was that. And then allocation concealment was through the varying block sizes, yeah. And blinding was only possible for outcome assessors. So outcome assessors and statisticians were blinded to the outcome. They didn't know which group. They just had a set of data from the accelerated group versus the other group. And it was not identified as such that who was in the accelerated. So obviously the outcome assessors and the statisticians can be blinded. And that was important because being an open label surgical trial, everybody knows, the patient knows, the investigator knows who's going within 6 hours to the emergency for surgery and who's going within 2 days or so. So that was clearly identified. Then we look at the statistical methods used to compare groups for primary and secondary outcomes, and the methods for additional analysis such as subgroup analysis, any adjusted analysis. For results, they want you to show a participant flow diagram, which clearly shows how patients were randomly assigned and how who dropped out, who consented, who then completed the study, and how the numbers at the end of the analysis are the same as the numbers who were randomized, so that the intention to treat is satisfied, that criterion. And for any group, losses and exclusions after randomization and why they were lost after randomization needs to be stated. And for recruitment, define the periods of recruitment and follow-up. So between 2017, March 2017 and April 2019, 21st, these were the periods of recruitment, and then follow-up was for 6 months after the closure of the trial, and the last follow-up visit was at this date. Why the trial was ended or stopped, that has to be stated. Baseline characteristics, so I think a lot of our colleagues have shown the baseline characteristics showing you the demographics through randomization, showing different factors, so that needs to be shown. For each group, the numbers analyzed, so the denominator included in the analysis. So obviously, whether it's a per protocol or intention to treat analysis, that would have to be stated. For each primary and secondary outcomes results, the estimated effect size. So whether it is in terms of whether it's a measure of association as relative risk or hazard ratio, whatever improve, you need to mention that. And its precision within 95% confidence interval. So throughout this day, last 2 days, we've been talking about a little bit about p-value, a lot about confidence intervals, and gradually you realize that confidence intervals are now in, p-values are gradually getting out of vogue, and but there's still people use it, so we still mention that. For binary outcomes, presentation of absolute and relative effect sizes is recommended, whether in a yes/no outcome. Ancillary analysis, any subgroup analysis, adjusted analysis. Again, I think Maham showed you the subgroup analysis where you had that forest plot, which showed that everybody on the left side of it, the dapagliflozin group, benefited on almost all the outcomes of interest in that particular thing that she showed. Then all important harms or unintended effects in each group. So obviously, as part of any clinical trial, it's important to look at adverse events and adverse drug reactions and report them, or serious adverse events and serious unexpected adverse events. All those things need to be recorded, and a quality assurance root cause analysis process needs to get into place. And if patients are harmed during a trial, then they need to be compensated, and that has to be documented within it at what level of compensation you're going to provide in terms of that particular harm. All important harms. So here my comments are that yes, they state clearly the intention to treat analysis. They do a subgroup analysis and interim analysis and methods to account for the loss of power. They mentioned they use this Haybital Peto method that I talked about. It states survival analysis using Kaplan-Meier, so it's the same stepped up approach and step down to look at time to event or survival from a certain point. So any event of interest. So when we talk about survival time, it's any event of interest that we have defined. It doesn't have to mean that people have to survive that period. It's just that it can be a balloon bursting after you blow it up, and when will it burst? Or if this like say a light bulb fusing off when the room becomes dark, anything can be looked at in survival analysis. So it's time to event basically. So you mentioned that, and then they also compared the survival rates using the log rank curves that again Dr. Maham showed you. The Cox proportional hazards, just to mention that here they mention it. Cox proportional hazards are simply like a straight line regression equation where you are looking at multiple determinants of an outcome, just like when you do a regression. So y is equal to mx plus c. So you can have a quadratic equation, y is equal to mx plus bc, bx plus c. So all of them determine with their slopes what proportionate risk or hazards are contributed. So if somebody is talking about showing survival curves with Cox proportional hazards and showing a difference, then in that curve also you can see that, okay, if the mortality was attributed to in one limb compared to the other a little bit more, then 40% of that was contributed by the fact that it was an emergency surgery. 30% was the fact that there were people over 70, 20% were that they were on anticoagulants, 10% were that they were immobilized. All those things can be proportional hazards can be determined through this. So they had all that. Then they talk about so when you're done with this, so when you're done with the results, then your discussions should basically just interpret what your results is, not overread into it, not underread into it, not be over speculative, but simply state exactly what your results are and how they are applicable to patients in practice, and what are the limitations and what are the challenges and what are the areas for further study. And you can easily state that in your discussion. So it's always nice to be humble here and talk about the trial limitations. What are the potential sources of bias? Selection bias, detection bias, analytical bias, whatever bias you can think of when you're reporting. And if you did multiple analysis, how was the loss of power attributed or accounted for? The generalizability of result to applicability of finding, with broad inclusion criteria and narrow inclusion criteria, whether it's a broad generalizability or narrow one, or exclusion criteria. If it's a broad one, generalizability may be decreased. So all those things need to be mentioned here while balancing benefits and harm. So you obviously want certain further interpretations, which either sometimes you do with number needed to treat and number needed to harm, or sometimes you let the reader go through that calculation. The trial registration, most good journals will never accept any trial unless you are registered in some form of registry, whether it is WHO registry or whether it is clinicaltrials.gov or ICTR, whatever. So registration number and what was your name has to be clearly mentioned on the manuscript. Where the full trial protocol can be accessed, that should be mentioned, and the sources of funding and the support, such as who were the sponsors, who was the suppliers, and what is their role in the study in terms of whether they were part of the reporting or part of the analysis, or were they not allowed to do that particular part. So in HIPAA conforms to the above guidelines, does not use speculative inferences. In fact, it was a negative study where they could not show, although other smaller studies and observational designs had shown benefit, but HIPAA could not show that within 6 hours of taking, although it made some intuitive sense that if you do surgery early, yes, it can be something maybe risky, but then the waiting period issues of thromboembolic complications may be decreased. So that particular aspect were not over speculated in this, and they comment on the limitations and generalizability and the fact that it was registered at clinicaltrials.gov and sponsored by the Population Health Research Institute of Canada. So if you look at your handouts and read the HIPAA trial, that out of all the 3 trials would be the most, the one which most conforms to the consort statement. Almost everything out there can be a good beginner's guide for how to report that. But then they also talk that you have to have a flow diagram. So anytime you do a randomization sequence and all, you need to mention what are the numbers of patients who are assessed for eligibility. And obviously out of those who were assessed for eligibility, those who did not meet eligibility criteria were excluded. So not meeting inclusion criteria, or whether they declined to, even if they met inclusion criteria, they declined to consent, and other reasons, all should be documented. And then out of those who consented and were able to be randomized, what were the numbers here? So this number N and the number analyzed here and number analyzed here, these two need to equal this number, randomized in our intention to treat analysis. So you have to say how many were allocated to this intervention, how many to that, how many were lost to follow up on both sides, and how many were analyzed in the end. And that is what the trial profile gives a bird's eye view of the trial. Similarly, in this HIPAA trial, just to give you an example, so that was a consort thing. The HIPAA reported it like this, that initially they started off with 27,771 patients who were screened, but eventually the ones who had eligibility criteria satisfied were 7,780. So then 2,970 were enrolled. So you can imagine that 27,000 patients were screened to enroll 2,970 people. So it's not a easy undertaking. Requires funding, time, effort, team, all those feasibility issues. And then half of them, or whatever the balance of randomization was clearly shown here. So some were lost to follow up, some were lost to follow up here. But as you see that exactly 1487 here, 1487 were analyzed, 1483 here, 1483 were analyzed. So what happens, let me just ask you a quick question. What happens if these patients, 8 patients who were lost to follow up in the rapid limb, and I don't include them in the denominator, how would the rapid limb look better or look worse? So listen to me carefully. So you have in the hip attack trial, one is a limb which is getting accelerated therapy. The other is a limb which is getting usual care. In the accelerated therapy, 8 people were lost to follow up. I did not include them in my analysis. Will hip attack in the accelerated therapy, this one, look better or look worse if I exclude them? 1,000 rupees, 2,000, 3,000, 5,000, 5,001, 5,002, 5,003, and I never had the 5,000. But here, so if you did not analyze those who were out, so you think they were the ones who performed well or the ones who had problems? Problems. So that particular group will look better or worse? Better. You will overestimate the effect in that particular limb if you did not include that particular thing. So that's why it's important to do the thing. So I think we are done, and we have 2 minutes before we have the certification ceremony. I think Professor Iqbal is here. He's already here. Oh, yeah. So just invite him. Until then, we can answer any questions. So we don't have any questions. See, I told you this consort statement will make you go to sleep. Check on them. Send them a message. Send them that I'm very upset and hurt personally, and they can give me the feedback at research@maru.com.pt. And I will give them my feedback.
Question to Dr. Ahmad is already answered. Is it ethical to change endpoints to be modified or change during the course of trial?
That's the thing. So when you change endpoints or change sample size, it's all part of some form of adaptive designs, and then you need to take them to the IRB to get an approval. So if you get the IRB to stamp it, they say it's ethical, you think it's ethical, but then I still don't like what they did in the remdesivir trial. They were selling that drug, and they couldn't show, they knew they probably wouldn't be able to show a mortality benefit. So they took a second degree thing, which was 4-day reduction in hospitalization. It's fine, it has cost effects, but not hard mortality benefit for that particular group in which it was indicated. So unless a subgroup subsequently showed benefit, which is fine. But so these are the kind of things people do sometimes to show an effect. So that was one. Any question from here, please? We have 18 seconds. Any question related to consort statement or the concepts? I hope it put together most of the things we've gone through. I hope it did, and we're going to be building on it subsequently in the next 2 sessions as well. So Professor Iqbal Chaudhary, I think, is on his way for distribution of certificates, and then we'll continue. So our program, what is in the end, last 15 minutes is being brought forward. So as soon as he comes in, we'll do that, and then we'll continue and add on that time to injury time. Any other question, comment, concern, complaint? So Dr. Abdul Rashid is saying that after food, there is no question. It's all digestion or indigestion. Thank you very much. Thank you for your attention.
Ladies and gentlemen, I hope you all are enjoying our interactive sessions with our experts under this 2-day training program on randomized controlled trials. Due to busy schedule of His Excellency, Professor Dr. Iqbal Chaudhry, we have requested him for an early closing remarks and for certificate distribution ceremony. With this, I would request His Excellency, Professor Dr. Iqbal Chaudhry, for his closing remarks.
أعوذ بالله من الشيطان الرجيم أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم السلام عليكم ورحمة الله وبركاته. It's not busy but a crazy schedule. See, in last 7 days, I traveled twice to Karachi, because came back to Lahore, went to somewhere about 200 kilometer, another destination. It's an absolutely crazy schedule. And I had a very good discussion with Professor Dr. Mushtaba Khatri, and I'm absolutely delighted that beyond this training course, we're thinking of big programs. And everything starts from a dream. And of course, dreams are extremely important, especially if you take them seriously and follow them. Whatever we see around in this country is, despite the government, is the result of crazy people, working very, very hard, pursuing their missions and visions and following their dreams. My institute in Karachi is also a result of dreams of great people like Professor Salim Siddiqui and Professor Taur Rahman. And what we intend to do is also a dream. For instance, yesterday we discussed that we're going to train 1,000 people to similar level as you have gone through an excellent training, 1,000 of them. So not a small number, but certainly a number which can really make a whole lot of a difference in a country like ours, in OIC country. So what we intend to do is to have in next 1 year, and for that we need to have great support from Professor Qadri and his team, that we would have 2 programs in Islamabad, 2 in Lahore, 2 in Karachi, 1 each in Peshawar, Multan, Faisalabad, perhaps 1 more place. So we'll have 10 of such programs. And then from here forward, we are planning to move to Africa and Central Asia. So we intend to do 1 program in Kazakhstan, 1 in Egypt, 1 perhaps in Uganda, 4 programs in Africa and Central Asia. In all such programs, we'll follow a hybrid mode, that people who can travel would come and participate in person of this excellent training which you have gone through. I'm sure this has really made a whole lot of a difference. And then these trainers, master trainers, would eventually train other people also. This is a great opportunity for us. You understand that perhaps if COVID was not there, nobody had ever asked us to conduct clinical trial. We were not on the map of clinical trial per se, we were asked to conduct clinical trial, not because we were known for it or we had any track record, because of a simple reason that they wanted to increase the number and they were looking for guinea pigs. So that has happened, but that has created a great niche for us, is a great opportunity that now we are known that we can do that and we can do it quite decently. This training and these trainings in future would make a whole lot of a difference, because you would be able to do things in a much better way by using the best of technologies, by using the best of ethical practices, the clinical trials which are internationally accepted. This is one dream, and hopefully we'll do that because we have sincerity of purpose and we have a great team. Second is that we at the OIC level intend to create a network of all those institutions and individuals who have conducted clinical trials, because we think that they're a great resource. They have gone through a process and they have all the potential to do that even better in future also. And any platform or a network can be an excellent mechanism through which we can learn from each other experience and engage constantly in a sustainable manner so we can learn from best practices of what is happening all around. Comstech mission is largely networking. I think the only thing which we do every day is networking. Provide you a platform where you can network and tend to know each other and then continue to work. Our work is largely to facilitate your meetings. We are doing it in Pakistan. We will do it in, we have done it in the past in every region of the OIC, from Suriname to Indonesia, and we intend to do it again as we go to post-COVID world. But this learning platform have actually provided us a great opportunity that even without traveling, we can still interact. May not be as effective as we create this informal linkages and develop friendship, but certainly better than nothing. So these two things are extremely important. And these are, of course, our collective dreams, and we'll work together. Each one of you can contribute because now you have reached to a stage where you have a good understanding of clinical trials and what are the aspects of it. Some of the finest people in this country, the people who know this field from inside out, or formally trained from the top institutions of the world, great understanding, have been your resource persons. And I'm sure that this is a resource which you cannot really get anywhere. I remember that we were establishing, my colleague, Dr. Raza, is perhaps not around, that we were at some stage, we wanted to establish our bioequivalence center, and I wanted to send people to Malaysia. And I wanted to send 3 people to Malaysia. And they sent me a bill, not Malaysian government or Malaysian institution, government institution, but a private company run by someone conducting bioequivalence studies, a bill of $500,000. And I said, good Lord. Of course, we spent not even $30, and we have used our network, and we were able to train them very well without spending much of money. And now we have very good quality people, even better than where they were supposed to go for training. So this is the kind of a network which can help us to promote, which can help us to move forward, and which can help us to learn from each other. Thank you very much, sir. Jazakallah khair. Thank you very much.
Thank you very much, sir. With this, I would request you to please award certificates to our valuable resource persons and international participants. First, I would request Dr. Abdul Rashid for the certificates. Ji, sir, please. सर आप दे रहे हैं डॉक्टर अब्दुल रशीद को. सर to Dr. Mushtaba Qadri. Dr. Waqar Hafiz. Dr. Waqar Hafiz. आप नीचे जाइए इधर. मिस खलूद जंजुआ.
Also a very non-pharmacist and clinical research fellow. He's part of our research.
Dr. Rahma Gilani. Dr. Mubin Iqbal. Professor Dr. Mubin Iqbal. Dr. Ahmad Fahim. Ms. Maham Zahid. Dr. Amaduddin Saqib. Dr. Shumail Anwar. Abduli Bajjan, our international participant. Baba Kunta Fofana. Amfal Badji. Manal Muhammad Al Sayyed. Mehdi Hassan Tushar. Rashid Saud. Dr. Khalid Shafi, one of our very senior participants here. Dr. Nagma Hashmi. Thank you very much, sir. And thank you to Dr. Abdul Rashid and Dr. Mujtaba. Yeah, and with this, we will continue with our technical program. And good luck, everyone, and hope to see you again in some other event, inshallah.
So, Bismillahirrahmanirrahim, I think if you can just take a stretching break if you want to stand up for 20 seconds, I have no problem with that. If you move your arm up a little bit, it's up to you, voluntary as well. And if you want to choose to sit, no coercion, full autonomy, you continue to sit. But normally, when you sit after lunch for an hour or 2 hours, Professor Mubin Iqbal can maybe vouch for it. Even maybe a small percentage increase in venous thromboembolism events in the right setting or no? Small percent.
It has not occurred so far.
It hasn't occurred so far, at least we can say that. So it's always good to stretch a little bit and to relax a little bit, and then so we can now hopefully get back to our schedule, and inshallah, Dr. Ahmad wants me to mention that there's a running tea, and if you go and have a running tea, he's going to miss his presentation. So don't do that, please. But if you want to go quietly and have running tea and bring it in, or just have it and come in, I have no problem, but as long as the number is not statistically or clinically significant of those who go out, okay? So You guys want to take a break of 5 minutes, or you want to you want to take a 5-minute break? Okay, I think we'll have to do that. So let's just come back at, it's 3:26 by this clock. 3:33, 7 minutes, please. Thank you. Thank you so much. Thank you for your patience, facilitators. I think it's we have to accommodate. Ketan regime. Coming to the last leg, but not the least, of a program that needs to now culminate in you being tooled with at least the definitions and certain concepts to be able to write a research protocol and budget for it, and to be able to then subsequently also be able to read and critically appraise randomized controlled trials and therapy articles. So these two sessions we've saved for last, but not the least. And without much ado, I'm going to introduce Dr. Ahmad Fahim, who is Associate Director for our Clinical Research and Research Department and Clinical Investigation Center at Maroof International Hospital. And he is a medical doctor with a PhD in biomedical sciences, and particularly related to pharmaceutical products and drug development. Some of his work has been in that area as well, cancer-related, cancer therapeutics. And he is a certified clinical research professional who has been working in clinical trials for the last at least, 7, 8 years as well, 8 years? 12 years, has worked with the pharmaceutical industry as well. And so he has an inside out view of the clinicians, the pharmaceutical industry, and as well as the actual clinical trial world, working with CROs and others. So he knows what it takes to convince bodies such as regulatory bodies and grant bodies and sponsors in how to put together a research protocol. So he's going to spend the next 30 to 35 minutes showing you how to develop a protocol and including protocol budget. So Dr. Ahmad Fahim, all yours.
Okay. Assalamu alaikum. Thank you for being here and staying there for so much. Thank you. Today my talk is on two areas particularly. My first half will be is sort of a more on the subject which is more academic, while the other topic actually deals with trial administration. So we are going to talk about protocol development, and the second half will be in covering budgeting in clinical trials. Okay, so this is usually what happens when we are thinking of we are working in clinical trials. We have a great idea. We're thinking that we have to work on. There are certain things which we have to look for. Do we have appropriate population? Then do we have the appropriate setup for doing that particular trial? With reference to setup, I mean, do we have that sort of expertise, that sort of principal investigators, that sort of trained research staff which can actually deal with clinical trials? Supportive clinical staff is really important, because if we aren't much trained in clinical trials, there will be lots of protocol violations, protocol deviations, and that will have a very deep impact on the results of the study and overall on the conduct of the trial. Then do we have the time and administrative support? Because if we don't have enough time, we probably cannot conduct trial well. Then do we have the relevant funding for that, doing that particular trial? And we have to deal with lots of legal and contractual issues as well, because if they are not dealt with, they will indirectly affect the conduct of the trial at various steps. Okay, so, okay. Tell me, why do we think we need a clinical trial protocol? Anyway. Just say anything. Why we need a clinical trial protocol? Okay, so I give you a scenario. I have a great research idea for which I developed a clinical trial protocol. Now I have targeted certain patient groups. Those patient groups are located in 33 different countries. Okay, so I have the relevant funding to do that particular clinical trial in 30 different countries. So I have 30 clinical investigators, and those in 33 countries, if each country has each single clinical trial site, then they probably might have clinical research staff as well. So do you recognize that for the safe conduct of the trial, everybody needs to be aware of what they're going to do? So this is what the trial protocol actually addresses. In 33 different countries, every person has its own understanding of doing research. But in trial, we cannot afford to have so much variations of for variations of experiences, because we have to stick to what the protocol says. So to predict and make allowances for the real world behavior of different individuals who will be involved in clinical trial, with the expectation that they will behave according to the protocol. So this behavior of according to the protocol is very important. It's in clinical trial conduct, we simply cannot accommodate any variation in the behavior. Okay, so as I said before, in this particular scenario, the protocol will be potentially used by many different people. They can be data managers, they can be statisticians, report writers, ethics committee members, and for most important, the study site staff, which include investigators, site study staff coordinators, nurses, pharmacists. So all they have to be on one page. They should have a consensus of what they're going to do in that particular clinical trial. So according to ICA GCP, a clinical trial protocol is that describes the objective, design, methodology, statistical considerations on organization of the trial. Usually, the protocol also gives the background and rationale for the trial. So there's another term which we very frequently use in research, and it's called proposal. So can you differentiate what a research proposal, and what's the difference between clinical research proposal and a clinical trial protocol? I think everybody who's related to research must have written some sort of research proposal, probably during their career. I'm sorry, it was not audible.
The ITs may be involved in the clinical trial protocol?
Okay, that's one aspect. Professor? I just want to listen because the clinical research I'll address.
Assalamu alaikum, sir. Sir, one thing is protocol and the other is you were talking about?
Research proposal?
The proposal. Sir, proposal is basically which you give for the IRB ethical committee before telling them. And the protocol are basically the guidelines you have to follow. Make sure the research is done properly according to the GCP.
Okay, I'll address. Both of these terms are often used synonymously. Okay, however, when writing a protocol, clinical trial protocol, particularly, we have some very, you can say delicate delineation. And it's technically a clinical research proposal is a rather brief document, which describes an idea for the project, emphasizes the research question and rationality of the research question, while a clinical research protocol is actually a detailed roadmap. So this is basically sort of, you can say, some differences. Grant proposal we usually write, and it's basically a sort of a proposal. So it's a scientific document, while a protocol is a regulatory document. So you can see the contents will be actually dictating by this. Okay, then in writing a grant protocol, its main purpose is to obtain funding. While in clinical protocol, which we are more addressing about putting every detail in that document. The focus of the grant proposal is to target aims, hypothesis, methods. While in clinical protocol, as we have discussed yesterday and today, there are very, very specific objectives, primary outcomes, endpoints, things like that. Feasibility for grant proposal is we're looking into cost, equipment, staffing, are they available or not? While in clinical protocol, we look for enrollment and implementation. So patient enrollment and implementation is most important in a protocol. Then a grant proposal is reviewed by a scientific committee, funders, potentially IRB, but not always. Like clinical protocol is always reviewed by scientists, sponsors, and IRB. IRB review is must. Then the tone of the research proposal or grant proposal is persuasive. While a clinical protocol is more informed and detailed, as I said, it's a regulatory document. Okay, so now we have to think of, okay, we are going to write a document and it has to be, we have to put every details in it. So how much details what has to be put in? Is there a way of taking a guidance, how much detail we can put into that protocol? So this is a matter of judgment with reference to what particular area we are going to work on with relevance to trial. Okay, so we first have to start mapping out the study, whatever our research question might be. This is just a general layout. This general layout is usually followed in most of the trials. Then, like we have visits, visit 1, visit 2, visit 3. Then we have to decide on first visit what will happen, on second visit what will happen, on the last visit what will happen. And this is how we're going to write every details. So by mapping out, we can think of, okay, with relevance to, if we have mapped, we have a layout of, okay, on visit 1, we'll be screening it. On visit 2, we'll be we'll be probably randomizing it. On visit 3, we'll be actually thinking of giving the experimental or the standard care drug, whatever it's assigned. So I'm going to give an example of HIPAATAC. This is a very good example. This particular paper is actually a published trial protocol, not the actual paper. This is a trial protocol which is published. There are no results in this particular paper, but it's a protocol which is published. So you can say that in good comprehensive trials, even protocols are published, even before you have started working. Okay, so you can use it as a template in later studies, but I'm going to give you this example. Okay, so they have given a work plan. We have a first box where they're mentioning that there'll be participant screening and recruitment, then there'll be participant randomization, then there will be an accelerated group and a control group. So this particular layout is giving classical example of what type of details we are going to put in every block. Okay, so this is a classical, you can say, headings with relevance to the elements of the protocol. These are certain basic elements, but we have a far better comprehensive guidance. There is another statement which is called a spirited statement. This spirited statement is actually a very standard 33-item checklist, which gives a very, very good idea, very, very comprehensive idea of what type of detail we are going to put into the clinical trial protocol. So as I said before, the general rule of thumb is we have to have a guidance of what type of detail we are going to put into the trial protocol. This particular spirited statement can guide you. There are other templates and checklists available as well on internet. Some universities have their own checklist, but if we do not have any particular checklist to follow, the spirited statement is a very, very good checklist to follow. They have very, very comprehensive headings. It's a very comprehensive statement with relevance to what type of details we are going to put into the clinical trial protocol. Okay, so I'm going to just give an example of the protocol portion of the HIPAATAC trial protocol. You can see that we have the number 24 as research ethics approval portion. Okay, so they have I've just taken a snapshot of a particular paragraph. Here in this particular paragraph, they have actually comprehensively mentioned what type of ethics they are going to, how their ethics approval has been taken, how the IRB has reviewed. Okay, at times this happens, and this also happened in the HIPAA trial protocol as well, that it undergone multiple protocol amendments. So every protocol amendment has to be reviewed by the IRB as well. So all that detail has to be also incorporated in the trial protocol if we are intending that there might be a protocol amendment. We can anticipate this as investigators at times that if we are ourselves not sure, there might be something, we probably might have some amendments. And usually they, in very, very large studies, very, very large sample size studies, we do have protocol amendments as the trial starts. So all that detail is mentioned in appendixes or annexures, and it's published. Okay. Then we have a dissemination policy as well, how the results will be disseminated. So this particular paragraph is actually supplementing also how the results were disseminated. Now, why they have mentioned it here? Because results are not just published in a journal. They have to be, the results probably have to be communicated to the patients as well, because in informed consent, patient might ask, sometimes it's mentioned in the informed consent that we will share the results with you. The patient might ask that, okay, I was enrolled in this study. Now tell me what happened in the later stages. What was the result of the study? So that's also a form of dissemination. The Data Safety Monitoring Board, the IRB, the regulatory agencies, all they'll be asking for results. So we have to mention how those results have to be disseminated at various stakeholders. Okay. So this was just a brief idea of how we are going to write a trial protocol. Okay, then we come to trial budgetary considerations. Okay, so can you tell me who prepares a trial budget? Anybody? Budgets are usually financial documents, so definitely some financial experts are there, but do actually they prepare trial budgets? Okay, good. But sponsors has certain people. Okay, so trial budgets are usually prepared by individuals who have worked in clinical trials at various stages. They're senior people. They're not junior staff. They're senior staff, and their seniority is actually judged by at what stages and how much experience they have taken in clinical trial with relevance to have they gone with various stages of any trial, whether it's related to recruitment, patient recruitment, or screening, or results interpretation, or study closeout, all these things. So these are those individuals. Our trial budget is not prepared by a single individual. They're a group of individuals. They're a group of people. So that particular group is experienced enough to gather data with relevance to what type of particular anticipation they can have with relevance to screening, with relevance to randomization, cost of randomization. I mean, what will be the cost of enrollment in a standard care versus experimental care? I'll tell you, there's a difference in both. So this is primarily an anticipation, but that anticipation has to be very calculated. Okay, so study feasibility questions are there to generate a budget. The person who has to prepare a budget has to understand the protocol, the required procedures, and the scope of work. Is this clinical trial feasible with institution's clinical research program? Now, this particular point I'm mentioning here is because at times we do have scenario where a sponsor comes to us and says that we want to do an orthopedics trial. But I'm working in an institute, and this might be possible that I'm working in an institution where probably orthopedics is not that much developed specialty. So is that feasible enough to do a clinical trial where I know that my institution do not have enough patients to do an orthopedics trial? Okay, then are the requirements of the clinical trial reasonable? Is the patient population available? Now, this particular point is happening a lot with reference to these days with relevance to COVID. There are lots of sponsor agencies. At times they say that, okay, we have vaccine, but this vaccine works, and probably we have to do experimental trial on a wild type COVID, wild type SARS-CoV-2. Now, wild type SARS-CoV-2 is not available at the moment. It's very difficult to be found because there are so much variations in the population. So if any sponsor comes to me and say, okay, I have, this is the amount, this is the funding I'm going to give you, and you have to prepare a budget where you have to work on the wild type SARS-CoV-2. I don't think so that it's feasible. Are the inclusion exclusion criteria too narrow? This is one example as, okay, is there appropriate study staff available to conduct the study? Now that's very important because these are the feasibility questions on which budget is going to be prepared. So it happens, why bother budget? The budgetary people are worrying before as well as afterwards. Okay. Why we are developing a budget? Because we have to understand the cost. As I said before, that it's related, this particular area is related to trial administration. So we have to worry about what can be the cost of each step, who will pay for what. There'll be lots of costs. So, and there'll be hidden costs as well, which we have to anticipate and presume even before starting the trial. What will be the cost of documentation? And believe me, in a trial protocol, in a trial conduct, there is enormous amount of documentation involved. Okay, identify research related to standard of care costs, and then informed decision. So whatever costs we are going to think of putting in for a particular trial, that has to be an informed decision. Okay. While preparing budget, this particular area that either it's a sponsor initiated or investigator initiated trial is very important because it's relevant to taking grants. We have a fixed amount of money and we have to think of a budget keeping in view of that capping, that okay, this is the X money I have and I have to accommodate everything within it. However, in sponsor initiated studies, we do not have that sort of capping. We just have to look into the idea and there are certain other things. And in sponsor initiated studies, we have more freedom to negotiate, not typically take it or leave it, but in grants, this is the case. At minimum, we need to cover the cost. This is the bare minimum criteria. However, but it's okay to do a little better than cost cover, like charging for hours for people working on it. Okay, so what to consider in a clinical trial budget? Difficulty and complexity of the study. At times, the study might be really good with reference to the funding and the idea and the budgets they are offering. However, the study might be complex. We don't have enough trained people to do that. Then the study preparation requirements, patient numbers. The patient numbers are one of the most problematic things to handle because at times we commit that yes, we can enroll this particular amount of people, and at later stages we realize that somehow something has happened, like the epidemics have happened. COVID epidemic has significantly disrupted the enrollment of trials already running in the field. Then inclusion exclusion criteria for subject recruitment and staffing. Okay, then there's invoiceable cost, pass-through cost, which include charts review, IRB fees, protocol amendments, adverse events, severe adverse events management. This particular area is important because who will cover for those adverse events and severe adverse events? So that has to be taken into account. The safety reports, audits, who will be going to be audits the sites. There'll be CROs, there'll be sponsors, there'll be regulatory agencies who will be auditing the particular clinical trial site. Who is going to cover them for their time and for their effort? Okay, then screen failures. Now screen failures are like something they have to be accommodated. Like previously during the hepatitis trial, as Dr. Kadri mentioned, there were 27,000 patients screened, and we ultimately take almost 2,700 patients approximately. So you can see we have to have that sort of funding for thinking of 27,000 patients and potentially probably for those screen failures as well. Okay, then shipping of samples if the study requires that, study staff time with monitors. Okay, so this is the same flowchart I have mentioned previously, just a few slides back. I'm going to just want you to anticipate the cost. Just look this particular slide with relevance to cost. So, okay, if I'm going to prepare a budget, I have two groups, accelerated care group and a control group. So for control group, and for accelerated care group, the costs of participant training recruitment will be the same. Okay. Then during randomization, probably the software pays equally for both, so it will be the same as well. And the resource persons involved will be the same for. However, once the patient is enrolled, for the accelerated care group, potentially everything is being managed in the ER. So ER has its own costs and own additional things to be incorporated as, because in this particular trial, we were thinking that in the accelerated care group, we have to do hip fracture surgery within 6 hours. While in the standard care group, we were thinking of doing surgery as the standard of care, which is around 48 to 72 hours. So within 6 hours of doing surgery, it means that all the anesthetic fitness, all the cardiac troponin levels, all those investigations has to be conducted potentially in emergencies. The cost is being raised significantly. So in this particular trial, the budget was like, for an accelerated care group, it was almost 3 to 4 times the control group. And for the rest of the levels, portions, follow-up, and analysis, and dissemination, the cost was almost the same. So you have to take in an investigator-initiated protocol, we have to take into account data management expenses, study-related personnel costs, equipment supply expenses, diagnostic fees, and then publication costs. Then in the start of the trial, there are certain costs which are, you can say, one-time costs. There are startup costs, which requires protocol review by investigators, study coordinators, because they're putting so much time in putting up that protocol ready for themselves. And site selection visits, site initiation visits, and off-site meetings. So all these are startup costs. Okay, then there is a detailed budget development. There'll be lots of people involved in it. There'll be IRB submission. Usually lots of various IRBs have certain fees because of the time and effort they're putting, the copies and amendments, advertising, patient recruitment, pharmacy protocol. So all these are just, you can say, certain headings. Then we have per subject costs. So those were the costs which were not related to the subjects, but like subject costs, initial enrollment costs, screening, which includes medical history, physical exam, additional tests. As I said before, that this might be possibility that in experimental group, there will be more tests or whatever the patients are randomized, there'll be extra tests for those people who are enrolled in the trial. Then there'll be screen failure. So all these have certain costs. Okay, then there are room charges and hospital beds, because if the patient applies and if the trial requires, patient might be admitted for a certain length of time. Okay, so, and there are certain recurring costs to consider as well, because usually trials run more than a year, 2 year, 3 year, 4 year, even 5 years, because patients has to be followed for certain time to record events. So there will be an investigator fees, there will be study coordinator fees, then there will be subject stipend. We might call patients to follow up, and they're coming not because of their own health needs, but because we are asking that because they are enrolled in the trial, they're coming to our setup, they have to be reprimanded because they're coming for that particular trial. So that is the subject stipend. Then there are general clinical research center services. In a small trial, although taking the CRO services is not mandatory, but in big trials, when there are lots of patient recruitment involved and and the trial is very complex, then it's suggested that people should take the clinical research center services. Then there are one-time costs. As I said before, there'll be RV preparation, publication fees, closeout fees. Okay, so the critical thing which we have to think of is that, as I said before, in that particular layout of the flowchart of the HIP attack trial protocol, is standard of care cost has to be, and the research related expenses has to be discriminated very well, because there is a very, very high chance of underestimating it. Okay, so this is a snapshot of a typical trial budget. This is just an example, just an idea how it looks like. Usually budgets are confidential documents. You can see that we have put certain amount at screening, then baseline, then there's a visit 1, visit 2, visit 3, and we have put everything. So the amounts are actually putting here by assuming the amount of services rendered by people, and how many people probably there might be people taking the informed consent, and they'll be putting their time and effort that hourly rate has to be calculated. There'll be inclusion-exclusion criteria review, and people will be reviewing that as well, that whether this patient is suitable for enrollment in the trial or not. There'll be medical history, there'll be demographics, there'll be physical examination, and then there'll be, as I said before, tests, specific tests to actually see whether this particular patient is relevant to, is suitable enough to be enrolled in the trial. So there'll be cost per visit, there'll be cost per subject, and there'll be then we have certain service charges regarding the center involved. These are some common budget mistakes we encounter. The most important is underestimating the time required for startup. Usually, when a trial feasibility comes to a particular center, usually, I know it's not a mandatory thing or it's not a rule of thumb thing, but it takes almost 6 months to more than a year to actually start the trial, because the IRB process is there. The probably protocol will be undergoing some amendments. Then there'll be site initiation, and IRB approval delay. So those are the things which actually then overestimating the ease of recruiting subjects. At times we realize that, okay, our OPD is full of patients, we can enroll trial. We can enroll them in the trial. That is simply not the case, because we have to understand this, that if we put a particular, if we are going to put the option of enrolling a particular patient in a trial, you might decline it, people might decline it, and then we have to think of that how much patients probably might be interested in being enrolled in a trial. Then there are other reasons as well. Okay, then usually we under budget the unexpected. As I said, that the budgets of all those, most of the trials were being run in the COVID era, and they have been initiated before. Most of them were in trouble because site recruitment was really trashed because of the lockdowns and all those things. Payment terms, this is a particular area and it has to be negotiated well. So there's a rule of thumb. It's that in clinical research, it always takes longer than you plan. So delays are there. What if we plan better? We can minimize that delay. And the takeaway message for developing a budget is one size does not fit all. Customized budget, taking study complexity and local realities into consideration. Every site in every country has its own ground realities, and those has to be taken into account while preparing a budget. If it's not in the contract, you're not guaranteed to be paid. So this happens because at times this can be just consider that I'm just giving an example that a particular site was targeted for enrolling 2,000 patients. That site enrolled 2,000 patients, and they signed the legal contract for those 2,000 patients. The sponsor wanted that, okay, this particular site has outperformed all of the sites, and they have to now, we can give them a new target for enrolling 1,500 patients more. Now that site enrolled 1,500 patients more without taking into consideration the additional financial costs. So as I said before, this can create trouble for that particular site, because now that additional 1,500 patients were not, the site hasn't signed an agreement for that. So this can create lots of discrepancies and lots of disputes. If you don't ask for it, you won't get it. This is what our experience is about, that this has to be negotiated really well with the sponsors or with people who we are working with. Don't let aggressive sponsors push you around. In the field of trials, these are the experiences. Use your institution resources to help with the negotiations. Good cop versus bad cop. Remember, everything has an expense. Thank you. Thank you.
Thank you, Dr. Ahmad, for going through a comprehensive protocol formulation exercise, at least the outline of how it needs to be done along with the budgetary exercise. And it obviously is a team effort, and it's important to let people who have had the experience or who understand financial figures take a look at it, because there are a lot of unwritten expenses that you may incur in your excitement to get research protocols at your center. Any questions online or on site? A couple of questions we can take each. on
So-
site, online?
Online, we have a question. How
Okay. Okay, this is a particularly important area. As I said before, that if we are experimenting new drugs or new clinical indications, this potentially might be the possibility that that particular trial subject might be having some sort of side effects. And usually they are covered. All the legal agreements, the site has to ensure that that coverage is there. But one thing is there. Whether it's an adverse event or whether it's a severe adverse event, it has to be very, very clearly and categorically mentioned in the agreements. And for that, sponsors and all those people who are regulating the trials, they look into that particular definition with relevance to that particular clinical trial protocol is very, very clear that what type of event has to be taken as an adverse event, what type of event has to be taken as a severe adverse event, and then what type of coverage the sponsor is going to do. And afterwards, that particular thing is very, very meticulously looked by the IRB so that the trial subjects may not be harmed or left over.
Yeah, so one should always try and make sure there's an indemnity insurance built into these kind of unexpected injuries that you're obligated ethically to address and to take it to the point of complete care. So any question on site? Yeah.
Thank you so much, Dr. Ahmad, for such a nice presentation. Sir, as he said that there are a lot of people involved in the clinical trial and budgeting, and so many jobs are being created, and statisticians and everyone, like many people. So I have seen recently in Pakistan, in Institute A, Institute and B and C, they were paying different amounts to the patients or the people involved in the research. A was giving 2,000 per patient, B was giving around 4,000 per patient, and C was giving 20,000 per patient. All of them in Pakistan, all of them sponsored by China or someone else. And so what is the criteria at which A is paying 2,000 and C is paying 20,000? Number one.
So
is there should be a rule for that?
yeah. So
Sir, another question.
Please go ahead.
From Dr. Abdul Rashid Saab. Yeah.
Please.
Why do we have different criteria for clinical trials in allopathy, homeopathy, and nutraceuticals? Why do we have different criteria?
Sir, Dr.
Yeah.
So
different criteria का है.
Sir, in English, please. So we have some audience, multinational.
We have different criteria because I think in WHO there is also different criteria for herbal medicine. There are different protocols, and for allopathic there are different protocols. In Pakistan, we are following ICH guidelines, also the WHO guidelines, and we have the experience regarding vaccines, biologicals, and also the allopathic medicines. Recently, in the last 2 weeks, we were working only on ventilators. We have developed our own protocols for regarding ventilators. But today, we are thinking about the clinical protocols for herbal medicines. Today, we're discussing with Professor Reza Shah. And today in my office, we're making a committee, one from Karshi, Hakim, one Hakim from Karshi Dawakhana, one Hakim from Amdad, one Professor Reza Shah, one me, and one also from Science and Technology. We have made 3. And we are going to develop the criteria for herbal medicine. as far as homeopathic is concerned, still we have not worked. And we'll take the experts from homeopathy, from the scientific backgrounds, then we'll make criteria for homeopathic, because still we ourselves, DRAP is in infancy. This we admit this thing. We are learning still now. As I mentioned that regarding clinical validation protocols of ventilators, we have given the approval of 2 ventilators, one from Atomic Energy Commissions and one from comstech, because the situation at that time was so bad that we were thinking, the INCOC and also the ministry were thinking, if COVID is increasing and the hospital will be full of patients, then what we'll do, and there will be shortage of ventilators. So without having any protocols, we have given approval. That approval has been conducted in CMH and also the General Hospital. Although we have given the approval for 96 hours, but actually those 2 ventilators, they work for 23 days. So regarding allopathic medicines, regarding herbal medicines, regarding allopathic medicines, we ourselves are in infancy. We have to take some biomedical engineers, pulmonologists, and intensivists, so many clinicians. Even I tell you, one of our retired Major General, he said, "I'm sorry, I cannot chair the committee of ventilators because this is we have no more expertise." So allow us to breathe in DRAP. So we go gradually. Thank you.
Thank you very much. Thank you. I hope that answers most of your questions, and we can discuss what you mentioned about the other negotiation aspect. We will now move on to what we have saved for you as a culmination of the learning activity through Professor Mubin Iqbal's expertise in the field, having spent time in internal medicine, pulmonary critical care, clinical research, as an educationist, as an IRB chair, all these experiences putting together, but it's actually his performance in the field demonstrated over the last 2 decades as an evidence-based healthcare practitioner that he is recognized, mashallah, in the country and also abroad. And from that experience, we're going to try and see if we can extract some further learning objectives from some of these trials that we had for you. So without coming in between him and you, Professor Mubin. Thank you.
Assalamu alaikum. Bismillahirrahmanirrahim. So, I was sitting and kind of trying to flex my brain muscle that what else is left and how can I make you concentrate for the last few minutes of this particular workshop. A disclaimer that first of all, whatever Dr. Qadri has said, just forget about it. Just think that I am a clinician, practicing clinician, someone who know nothing about randomized controlled trials in terms of doing them. But at the same time, when I see patients, I ask questions. I question my way of practicing. So when I do that, I always come up with questions that I don't know or my team does not know. So I need to find answers to those questions. And you know that in order to find the answers, you need to develop that question into a certain format that you have learned. Can you name that format? Pico. So I need to figure out, okay, patient, intervention, comparison, and outcomes that I am looking for. So once I do that, then I go ahead and figure out, okay, so I add another small letter at the end of that Pico, which is called T. Or we can also call it as D. Could you guess that? T or D? Okay. I did not hear, but did you say something? G?
Treatment.
Okay, so yes, T for treatment, T for therapy, D for design, whatever you want to call it. So I need to know that the type of question that I have asked, I need to figure out what is the best design to answer, study design, to answer that particular question. And most of the questions that we ask at the bedside, point of care are going to be about therapy. And you know that what is the best design for a therapy question to find the answer? What is the best design that can answer your therapy question? What is the best study design that can answer your therapy question? You have spent 2 days. Randomized control trial. So randomized control trial, by definition, is primarily meant to find your answers pertaining to interventions, therapies, right? So you generally, you don't use it for diagnostic questions. You don't use it for prognostic questions. You use it for interventions. So that's what you are trying to learn. That is one thing. Second thing is, I am not a statistician. That's a second disclaimer. So when I say that, that means that I don't know about single tails. I don't know about two tails. And I lost my tail several million years ago. So forget about tails. So I'm not going to put you to a lot of statistical issues. But one thing, if you can notice on this particular slide, which is going to be the perspective that I want you people to take with you, is on the right top corner. And if you can just follow that small logo, I'm not sure whether you can read that or not. It says, even I can't read because I don't have my glasses on. You can say validity, results, and apply it. Just remember these three words. These are so important concepts. And let me ask you another question. You have gone through 2 days of pretty intensive, reasonably intensive work on a randomized control trial. Whatever you were trying to learn in terms of designing, conducting randomized control trial, right from the ethics all the way down to conducting it, results and all that stuff, and statistical analysis and whatnot. What do you think you were, why do you think you were trying to do all those things? Why do you think it was so important for you to know about randomization, blinding, concealment, intention to treat, and what not? Why do you think it was so important? As a researcher, why questions are the real ones. So why do you think this was so important for you to know the methodology, and that too in a fashion which is probably gold standard, right? Why? Sorry, to
have
accurate results.
Yes.
Absolutely right. And what is a better word to say that? Valid results. So number one, validity. So you were trying to make sure that whatever you are going to do to have a wonderful internal validity, quote unquote, right? Because this is all within the facet of a randomized control trial. So if you don't follow any of these steps, what is going to happen? Your results are not going. Now, let me ask you another thing. which is a partially statistical concept, but we should be able to say that even in literal format. What are we trying to minimize by achieving internal validity? Error, risk. A better word for error starts with B. Bias. Thank you. So bias, systematic error or bias, they are the same thing. So we were trying to make sure that you were trying, whenever you design a wonderful randomized controlled trial, your bias remains minimum. You won't be able to get rid of it, but you can minimize it to the extent that your results can be reliable in terms of applying it. So that's one thing. So validity, results, and third concept we are going to discuss in a second. So what, why, and how of critical appraisal? So as a clinician, I would like to know that my question is going to be answered by a randomized controlled trial, but I need to know whether this particular randomized controlled trial has enough internal validity so that I can rely its results. And how do I do that? By analyzing for internal validity, evaluating the results to see whether they are precise and they are clinically significant, besides being statistically significant, and finally applying it to my patients. So that's the purpose, that's the perspective, which is from the side of an end user. So we are, as clinicians, we are end user of that quote unquote research, or the better name for this is, starts from E, evidence, right? So we are trying to use evidence to sort out problems at the bedside, right? So that's the way we are trying to do. So analyzing, evaluating, creating an applicable conclusion, and all this is critical appraisal of randomized controlled trial that we'll be mentioning a little bit. So the same things which I was describing are mentioned here again, that does this study that I have come up to answer my question addressed a really clearly focused question, PICO. The outcomes are patient-centered and they are important. Study used valid methods, and we are going to talk in a second, just a repetition. And valid results of this study are important, clinically important. As Dr. Qadri was mentioning about remdesivir, that initially they were looking for mortality, and they figured out that in order to change significance, achieve clinical significance in mortality, but that probably won't be easy to achieve with that kind of sample size. So they came out, okay, fine, let's just change the outcome to morbidity in terms of length of stay in the hospital. Are these valid, important results applicable to my patient or population? So important, because not every evidence may be applicable to your context. For example, I am practicing in Pakistan, you might be practicing in another country. Even within Pakistan, I may be practicing in an urban setting, a village setting, or any other setting. So all these settings can have their own intricacies. They are different kind of settings. So evidence, unless it is contextualized, has no meaning. That's another important perspective that you need to understand. So when I say internal validity in RCTs, so can you list them starting from your first lecture or session? So things that you do to achieve internal validity of randomized controlled trial. Randomization, asking a focused question, randomization, allocation, concealment, blinding, masking, blinding, appropriate statistical methods, intention to treat analysis, right? And what else? Results, are they precise? What are the measures of association? How narrow are the confidence intervals? Are they important for my patients? So these are the different steps, and exactly those steps we are trying to figure out, even when I am looking the randomized control trial from the perspective of using it for my patient at the bedside as an end user, not as a conductor of experiment, as an end user. So, so that is what we call internal validity. And as you can see, the purpose is to reach close to the truth, right? And you know that, I think Dr. Rama showed you 2 by 2, that there is a truth that we don't know, but there is something that we are doing in terms of experiment, and then you have false positives and false negatives, and then you have true positives and true negatives, right? Type 1 and type 2 errors. So you are just trying to figure out that we are trying to reach to the close truth, which is no one knows about that truth, but you are just trying to close to that truth so that you can apply that in some kind of confidence. And that confidence comes from confidence interval. Achieving validity in real life. So fine, experiment is done. I have reviewed an article, and article has something very important for my patient. But how to figure out that particular evidence is applicable to my patient? And that is what we call external validity, applicability. Remember the 3 things that you are looking at: validity, results, and application. So external validity depends on right patients, the cost of intervention, cost of maybe follow-ups as a result of use of that intervention, side effects, compliance of patients, compliance of physicians, availability, interactions, and the kind of outcomes that we are looking for. So in other words, it is not that easy. You just cannot pick something from somewhere else and say that it is applicable to my patient. It is difficult. You won't be able to do that, especially if you are sitting and working in an environment where policy decisions are made through National Health Service, right? They are the ones who provide medications, the cost, and a beautiful example is use of tocilizumab. Those of you who are providing care to COVID patients know that tocilizumab is a medication which we use sometimes to stop the storm, the inflammatory storm, right? And look at the cost, the availability, and the actual benefit which a patient is going to get. If you are not aware of all those things, you will be just prescribing patients blindly tocilizumab, and the family is going to go crazy because it is being black these days, and the prices have gone up tremendously high. So the question is, are you aware of its benefits? And if you are aware, do you know that this particular patient may not be able to afford that? And the kind of anxiety you are provoking in that patient's head or patient's family is going to drive them nuts. So important to know how to contextualize the evidence. So basic structure of an RCT, you guys know very well, angles. You know that you have a target population and you have your criteria for selecting them, then you have your sample, then you have your randomization, then you have your parallel groups, and then you have an outcome, good or bad outcome, and this is kind of a very basic structure. This is for someone like me who doesn't know much about RCTs. And then in order to use things in a more systematic fashion, if you look at this CEBM, how many people are, they know about Center of Evidence-Based Medicine? Oxford, Toronto, there are 2, and they are interlinked. So just go to Google, just write CEBM Toronto or Oxford. You will find a lot of learning tools about critical appraisal. So you can download them. And I think there was one included in your file as well for randomized control trial. And it also asks the same 3 questions: validity, results, and applicability. So if you look at the RCT appraisal sheet, if you have it somewhere, it's
hand out at the back.
Yeah, it should be somewhere. Understand question being answered, methods are valid, analyze results in terms of significance, precision of results, and then develop your own strategy to apply those results. Once again, the same things again and again. It is there in your handout. So we have already discussed this, and now, in fact, you know more than me about all these steps, right? You agree with me, right? So that was the article that has been referred to so many times. Dapagliflozin in patients with chronic kidney disease, published in New England Journal of Medicine. We have been using it as a good example of a trial which is reasonably good when it comes to categorizing in terms of high internal validity. So if you read this, this is our PICO of DAPA study. So I just put like P, I, C, and O. So you can, it's more of a narrative form. And if you read it, that's pretty simple. So you have a patient population, CKD, with or without diabetes. You have an intervention of DAPA. You have a comparator, which is standard therapy with placebo. And you have an outcome, which is primarily chronic kidney disease worsening in terms of GFR, mortality, or renal or cardiovascular outcomes. So are the results of the trial valid? So the same question. So the questions in that particular sheet that you have are, was the assignment of the patients to treatment randomized? So answer is yes. Were groups similar at the start of the trial? So how would you figure out that the groups were similar at the start of the trial? Based on you have the trial right with you. If you look at the trial, can you tell me that the groups, when randomized, were equal in terms of basic prognostic indicators, that they were looking known and unknown? How do you know? Very good, excellent. So table 1 is the one which gives you that idea that both groups are very nicely balanced. And that table tells you that randomization worked very well. So that's a way of very simply knowing. So if the last column, if you don't see any a mark that's saying significant, statistically significant, that means that it has a very nicely balanced randomization. And in this case, that happened. Were groups treated equally? Once I randomized to 2 different arms, subsequent follow-ups, treatments has to be same. If they are not same, what are we introducing? If they are not same, what are we introducing into the study? Bias, systematic error. And when you do that, the results are not going to be valid. Thank you so much. Were all patients who entered the trial accounted for? What are we talking about? Lost to follow-up. People who are gone. By the way, studies have shown that people who are lost to follow-up, they tend to have good outcomes or bad outcomes? Bad outcomes. So that is why it is so important that loss to follow-up should be accounted for. And were they analyzed in the groups to which they were randomized? What do you call that? What is the name of this concept? Intention to treat analysis. So when they are randomized to one group, they will always be in the denominator of that group. They cannot cross to the other arm. Even if they get the treatment somehow for whatever reason, still they will be part of the denominator of that group they were inside. If that is not done, what is compromised? Short of validity. Yes, validity, yes. But another concept of your RCT is compromised. No, I know, but there is something else. The very basis of RCT is compromised if your patient is included somehow in the other denominator. Randomization. Randomization. So that is why this is so important. You don't want to do that. So that's why it's so important. Okay, and then of course blinding, and blinding is important for you to know that blinding is not necessarily of patients, care providers. It can go beyond that. Generally, we talk about double blind trials. It can be triple, 4, 5, or more than, sometimes more than that. So what else? Who else can be blinded? Patients, care providers, investigators, okay, statisticians, outcome assessors who are going to decide that, especially if the outcome is non-mortality outcome. Mortality is easy to say, he's dead. but other outcomes are so important. So outcome adjudicators or outcome assessors. So there are so many, 4 or 5 different people who can be blinded. So it's so important to know once again. So DAPA in patients with chronic kidney disease, these are the inclusion and exclusion criteria. Why do you think inclusion and exclusion criteria are important for a practicing clinician if I have to use DAPA in my patients? Why? So important. So if my patient who has CKD with or without diabetes, with a certain kind of GFR, but with underlying systemic lupus erythematosus, those patients were excluded from this trial. So important to know inclusion and exclusion criteria because of applicability. Otherwise, in real life, sometimes applicability becomes so difficult that you start using medication left and right to different kind of populations which are not studied. But that does not mean that those populations should not be studied. Yes, they should be, but subsequently in the proper systematic fashion. And these are the trial logistics. 386 sites in 21 countries. Supply of medications was by AstraZeneca. They looked very similar, both placebo and the medication or active ingredient. Taste, color, smell, everything looked very similar. Monitoring was through an executive committee and an independent data monitoring community. Adjudication of outcome was through an independent committee whose members were unaware of the trial group assignment blinding. Trial funding, trial governance was through an executive committee, which had, I think, 9 members, and 2 were non-voting members from sponsors. So important to know. Otherwise, if sponsoring sponsors have the vote, then you know that what can happen, right? Because someone was mentioning the sponsor trials, they have higher element of sponsored trials as compared to non-sponsored trials may have higher element of bias, B. Okay, good, excellent. So outcomes. So primary efficacy outcome, you know that there is always a primary outcome. And primary outcome is so important because there is something that is calculated based on primary outcome. And what is that calculation that you do before starting your trial based on primary outcome? Sample size. Sample size is always through primary outcome. It can be a single outcome, but when you have that kind of an outcome, what do you call that? Composite, in which 3 or 4 things are combined together to make an amalgam. Things become very difficult at times to interpret when you have composite outcomes. So be very aware about composite outcomes. So these are, the primary efficacy outcomes. You know all very well what was looked at in terms of DAPA trial, and there were some secondary outcomes. And you know that the trial is generally not powered for secondary outcomes. The trial is not powered, but sometimes you can get some very interesting information which can be so helpful that you can even apply that in terms of confidence intervals and point estimates, that the secondary outcomes can still be used if they are very tightly if they are very tight in terms of precision. So power calculations, I have no idea. P-value subgroup analysis is important because you are going to see subgroup analysis. Only those trust those subgroup analysis which are planned when? Before starting the trial. You hypothesis priori, right? If you don't do that, what will happen? And most of us doing research, at least maybe should I call it developing world, we go on what we call fishing expedition. So, but we just keep doing something to find what? P-value less than 0.05. When you do that, unfortunately, the more hypotheses you test, the more is the chance that you will end up getting a p-value significant, which is by chance. So remember that as well. So, so some basic questions. Randomization was done. It was stratified, mentioned in methods. Concealment through computer web, mentioned in methods. Blinding, more than double. In fact, it is mentioned in methods. Placebo control, yes, we know. Mentioned in methods. Baseline table 1, you have already seen that. Very nicely done. Intention to treat, yes, done. Methods in mentioned in methods and statistical analysis. These were the basic baseline characteristics, and it's a wonderful gauge of successful randomization. And you can see, if you see the last column and don't see any asterisk or any pound or any sign, that means that none of the variable was statistically significant. So what are the results? How large was the effect? How precise was the effect? And those are things that and the precision is through which measure? Precision. Okay, so precision. So whenever you have a result is going to be in the form of a point estimate, and that point estimate is going to be one of the measures of association which Dr. Rema was describing before, and we relative risk reduction, and absolute risk reduction, numbers needed to treat, etc. But it is the spread, it is the confidence interval that gives us the precision. And this is just a kind of a simple recap of what you already know. By the way, who's from public health? There are people from public health, right? So public health people would like to call risk as something else. In randomized controlled trials, risk can be called as something else, especially for public health people. An occurrence which is happening for the first time is called as, in a group of people, population, incidence. Yeah. So risk is also incidence. So in these trials, you can in fact come up with incidence. Relative risk ratio, rate ratio, they all are the same. Relative risk reduction is 1 minus RR, and we'll talk about in a second. Absolute risk reduction is the difference between two risks. So risk in the control group minus risk in the experimental group. So, and NNT is just reciprocal of absolute risk reduction. Numbers needed to treat to prevent one outcome. And numbers needed to harm to cause one harm. So I need to treat so many people to get one harm. Warfarin to so many people will cause GI bleed in this much people. Number needed to harm. Warfarin in so much people will prevent this much strokes. Numbers needed to treat. So balance is so important. If the balance is more towards harm, you try not to use medication. If the balance is more towards treatment, NNT, you try to use it. But there is another thing that we are going to plug into this in this equation later on, who is going to be a major decider. So how precise are the results? Sample, target population, then you have a sample, then you have a point estimate, then you have a confidence interval, then you go back and apply to that particular population, because confidence interval gives you an idea that the results that you are getting are going to be applicable into that population which you are going to use them. So if someone has a point estimate of relative risk 0.76 with a confidence interval of 0.5, lower bound, upper bound is about 0.9. So you are 95% sure that another sample from the same population is going to fall within this range. That is what 95% confidence interval is. So another sample will fall into this range. So if they are tight enough, you can say that I can use that results with a good amount of confidence. So DAPA has a median follow-up of 2.4 years. It has very good adherence in terms of follow-up. Loss to follow-up was so little, and everyone was accounted for, and everything goes in favor of good internal validity. And these are the DAPA results. So look at the primary outcome. Primary outcome had a hazard ratio of 0.61 for all practical purpose. Remember, bedside collation, no knowledge of statistics. I have no idea what is relative risk, what is hazard ratio. I only know that hazard ratio is just like relative risk. So 0.61 means that 1 minus hazard ratio is equal to relative risk reduction, right? 40% reduction in primary outcome if I use DAPA. That is so simple to know. Forget about how hazard ratio was calculated, what was Cox proportional hazard model, and all that stuff. Absolute risk reduction 5%, 5 per 100. So treat 100 people with DAPA. How many are going to be benefited? 5. You treat 20, 1. This is numbers needed, simple mathematics. And then you have a confidence interval of NNT, which is 15 to 27. Pretty good, actually. Pretty good. So, but over a period of 2.4 years, at least median. That's so important that when you are thinking of using an evidence on a patient who is sitting in front of you, you need to know that how long are you going to treat that patient for. It is not a matter of 4 days, 5 days. So patient should be able to sustain with that medicine in terms of affordability for at least 2.5 years before you can safely say that one primary outcome is going to be prevented, right? So it's so important. And these are the curves that you already know. Composite endpoints are so important. Composite outcomes have issues, and when they are joined together, amalgam, so they have issues. So you need to know that are they biologically linked? Are they of equal value? Mortality versus drop in GFR. Mortality is a big thing. Drop in GFR may not be that much as a surrogate. So always think about equally important. Mortality might be relatively rare. Drop in GFR is more frequent. And as you can see this particular trial, you will see that individual outcome evaluation is so important. So, and I think if you go back, you will look at these things. These are subgroup analysis. The only one thing which I wanted to see that the Asian population was touching the line of no effect in DAPA trial. That's an important conclusion because you'll be using this particular medication in your Asian population. But it was very safe medicine. All the safety outcomes were in fact better in DAPA as compared to the other group. And when it comes to applying the results to your patients, you need to figure out that your patient, is it the same as the patients who are in that DAPA trial? And all the clinically important outcomes were considered in terms of outcome, in terms of mortality and all that stuff. So if I come back and start looking at, so my patient is someone who is very similar to study who has CKD, who may not have a lot of awareness about his disease or her disease. He or she may not be compliant because she lives in a far off place where sometimes access to medication can be an issue. Medicine can be available, but access can be an issue for 2.4 years. It's about 20 to 30 rupees for a 5 milligram tablet, and the dose is about 10 milligrams. As a provider, my access to the patient can be an issue as well. And I don't know what my patient think about it. So meaning thereby that when I apply an evidence to my patient, I need to be very sure that the decision making involves the patient, his or her circumstances, my own circumstances as a physician, and patient's values and preferences. So that is so important. And because the importance of this particular thing gets incorporated into a paradigm, which I was trying to come, where evidence is not enough, physician is not enough, and patient in isolation may not be enough to make the decision. So all these three components, they have to come together to have some kind of a decision-making process. And do you know what you call that process? Once these three things, evidence-based medicine, evidence-based healthcare, evidence-based decision-making, whatever you want to call it. So this is just a self-reflection exercise that once going through the last 20, 30 minutes, please have a look at it and start thinking maybe that way. Some resources, Gordon Guide, David Sackett, they are the champions in evidence-based medicine. Sackett passed away a couple of 3 years ago, in fact. And these are the references for this thing. Good luck for diving deep in evidence. Thank you so much.
Thank you, Professor Mabeen. Profound, because after all that research and all that detailed methodology, it all comes down to the patients. It all comes down to the population at large. It all comes down to healthcare and to improving outcomes of patients that we take care of, which is how research has to be looked at. So in the interest of time, I'll allow one question from here, one on the online. You want to have one comment from online, please? Any question on site? Any question on site? Yeah.
Oh yeah. Answer is yes. At least an outcome, because the trial cannot function without an outcome. Yes, you may not need any secondary outcomes, but you need to have one primary outcome, because otherwise your trial cannot even go any further. So yes, you need a primary outcome. Yes.
Thank you. Any question from here? Yes, please.
No, there is no difference when it comes to randomized controlled trial. So risk is something which is not happened, but will happen with the use of medication. And anything that happens for the first time during a certain time period is going to be incidence. But generally we use the term risk. And incidence is generally left for cohort designs as compared to randomized controlled trials. But by definition, it is incidence.
So, yeah, please.
You're right. What you are asking, that's right.
From where the doctors in Pakistan, they get information about different medicines that they have to use this.
Yeah.
Because I remember when in fact, Dr. Ranjit said that you should take chloroquine or levotine, Pakistani people, or like anything else in the world.
That's right.
Ivermectin also. They
Yes.
took steroids, dexamethasone also. They took
Azithromycin. Azithromycin as well. You're right. You're right. I think that's you're absolutely right. Case series or case reports or case observations, unfortunately, they are not very valid designs in terms of applying evidence. But in cases where you have pandemic situation where randomized controlled trials may not be available soon, and remember that we all know that when we started, we had no idea what is going to be working. Just anecdotal reports that azithromycin might be working. So yes, you can use that. And that was what happening all over the world. This is what we were doing. This is what US people or UK people were doing. But after 6 months, we had enough evidence that hydroxychloroquine, azithromycin, those were not a great. So comes out that if you don't use it in emergency situation, and there are some reports that there might be some benefit, sometimes I think it will be difficult for you to ethically justify not to use it. But once you have enough evidence, then everything just disappeared, including our method.
Okay, with that, we come to the end of this particular 2-day training done by Comstech, Maroof, and Drug Regulatory Authority of Pakistan. And we thank you for your presence online. We thank you for your presence on site. We thank all the facilitators, all the administrators, the hosts, everyone concerned with this activity. And we hope, inshallah, pray that Allah subhanahu wa ta'ala bless this effort so that you can use the ilm for the benefit of humankind, which is ilman nafian. That's real translational knowledge as far as I'm concerned. So God bless you all. Jazakumullah khair. And I know why you're waiting. There are some certificates here in my hands, so which I still have not been. That's just a small comment. So we'll just go through this quickly. Dr. Rabia Riasat, your certificate is here. If you can just come up and pick it up. Mr. Mufassir, if you can do the honors. Thank you. Samreen Fatima Sahiba. So that's the second one, please. If she's coming down, so just give it to her, please. Thank you. Huma Sahiba. That's the third one right here. Rabia Raza Sahiba. Thank you. Rabia Raza Sahiba. Dr. Aisha Wajid. Hassan Imtiaz Sahib. And they really like Hassan Imtiaz because there's another Hassan Imtiaz here. Hassan Imtiaz. Maybe they printed it wrong. Muhammad Tawab Khalil. Muhammad Tawab Khalil. Oh, there are two Hassan Imtiaz. Dr. Umar Yunus. Dr. Umar Yunus. Aisha Zaid. We'll have to correct it also. Aisha Zaid. Dr. Hassan Mumtaz. Aisha Zaid. Dr. Hassan Mumtaz. Dr. Hassan Mumtaz. Dr. Muhammad Asghar Khan. Dr. Khawla Shah. Dr. Khawla Shah. Dr. Isaac Asher. Dr. Isaac Asher. Sayyid Aqil Hussain Shah. Sania Umar. Sayyid Aqil Hussain Shah. Sania Umar. Sayyid Bulal Hussain. Muhammad Saad Zafar. Muhammad Saad Zafar. Sadia Birjis. Mustafa Mahmood. Sadia Birjis. And last but not the least, Sayyid Mufassir Shah. Thank you. Alhamdulillah. So thank you everyone. The buses are leaving for those who are going home in Islamabad. The rest, you have your evenings free. All the best. And tonight at 9 o'clock sharp, inshallah, I'm again going to come online and do the MCQs related to what we had yesterday. For those who missed online, I'd be happy to do that with you. Thank you. God bless you all. Assalamu alaikum.